الموافق 1980 تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
يخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
تخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، وسنقدم نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
السلام عليكم ورحمة الله وبركاته. الصلاة والسلام على أشرف رسول الله سيدنا محمد صلى الله عليه وسلم. Welcome, dear guests. I would like to welcome you for the Day 5 Biotechnology session of King Fahd Medical Research Center first conference. And today we will be exploring a very nice series of talks covering most innovative recent applications of biotechnology. And I would like to start today by introducing our first moderator, Dr. Nabil Al-Hakami, who holds an associate professor position of pharmaceutic at Faculty of Pharmacy in Abdulaziz University, an influential innovator in the global of biotech ecosystem, known for bridging scientific discovery with commercial success and with a PhD in pharmaceutical chemistry from the University of Kansas, USA. And he combines academic excellence with entrepreneurial leadership as the founder and the CEO of AI Terakia Pharma, Teriak Pharma, and many other startups. His work spans biotech venture capital, strategic management, and product commercialization, earning him the best Biotechnology Startup Award in Saudi Arabia in 2021 by F2 2022. Welcome, Dr. Nabil Hakami. I would like also to introduce our second moderator for today, in session 1, Dr. Ayman Bokas, who holds an assistant professor in the Department of Medical Laboratory Sciences at King Abdulaziz University and laboratory supervisor at Center of Innovation in Personalized Medicine. He is qualified with a PhD in medical genetics from University of Leicester, UK. Dr. Ayman specializes in medical genetics with research interests in genetics and epigenetics, particularly focusing on small RNA expression and its role in gene regulation linked to various diseases. Welcome, Dr. Ayman.
Assalamu alaikum wa rahmatullahi wa barakatuh. I would like to introduce Professor Martin Hassanein. He is the Vice President for Research and Innovation at Faqih Care Group, a large private healthcare provider with a comprehensive services and leader in academic medicine in the private sector. Everyone knows Faqih Group. He is also the founder and managing director of Saudi Vax, a pioneer Saudi biotechnology company responsible for developing and manufacturing high-quality vaccine and biopharmaceuticals. He's a professor of surgery and also solid organ transplant surgery consultant and joint professor at the Department of Pediatrics at Baylor College of Medicine. بس أنا ما أقدر أوقف على اللي مكتوب هنا. أنا know Dr. Mazen for a long time. If you want to know someone in Saudi, he's start from scratch as entrepreneur in the healthcare. He's one of the best you'll see in the kingdom. And he start from scratch till he built the Saudi Vax as one of the pioneer healthcare company in Saudi. And his visionary and all his career in the last few years, with all his experience, I always learn from him a lot of things about life, about career, about science. So it's a really valuable lecture, inshallah, will be. Thank you, Victor.
Inshallah, I'll be happy to just said. Thank you very much for the organizing committee for the invitation. This is really an honor to be here with you today. Biotechnology is the dearest to our heart, the best for last. So thank you again for the invitation. Today we're going to cover a few things related to our the major topic, which is what's the role Saudi Arabia can play. لو في ميكروفون أحسن. What's the role Saudi Arabia can play in the field of biotechnology? And I think before we jump into biotechnology, I want to let you know a little bit about the people outside of the biotechnology sector. How did they get introduced to biotechnology? This is very important. And the decision of jumping into biotechnology is a collective decision, and not only the group that is interested in the topic who make the advances on their own. Everybody else around you needs to agree on what you want to do. This is important when you go to the national level. If you want to go to the space, not only the space scientist needs to be engaged, a lot of other people need to be engaged so that the right committee, the right structure, the right organization is in place to allow you to grow. Biotechnology was introduced to our non-biotechnology people during COVID-19. And I think this was a major introduction because there was an awareness of where biotechnology played a role in the therapeutics and diagnostics capability during the COVID-19 outbreak. And so the public, if you go today to a public, even high educated individuals within the public sector, and you ask them about biotechnology, they'll probably remember three things: the diagnosis and swabs, and COVID-19, and the genetic testing, and vaccine. Actually, they might not remember the monoclonal antibody that was developed for COVID-19 therapy. So, which is good. It was a good awareness for biotechnology. It was good awareness for the public. However, there's a negativity also because this happens once in a lifetime. We're not really wanting to see another outbreak, although we know it will happen. We don't want to wait for that. While actually biotechnology is how things I learned in medicine, and not so long ago, I don't want to call it very long time ago, but we've learned a lot of things in medicine where are actually now absolutely obsolete. So we had certain genetic diseases that were incurable. Now they are, at least they're controllable. We had oncology cases that were futile. Now we have 5 and 10 years survival. We had autoimmune diseases that was absolutely no remedy for it. And now we control them. We have neurodegenerative diseases that we can control. We started to, when we used to diagnose certain diseases, it was just the simple pathological diagnosis. Now we have a subspecialized diagnosis based on additional new markers. And all this is biotechnology. And I think as a start, I think this is a good platform to start to let the public aware. COVID was just part of the picture of biotechnology. What else happened in the healthcare is a big part of the biotechnology. Another disruption that is happening, and we need to be also and make people aware of. We used to also separate very clearly demarcating biotechnology from med-technology from health technology. And so it was very clear, if you are med-technology or medical device and diagnostic, then that's a different route than biotech, there's a different route than health tech, which is mostly IT and digital. This is also getting eroded with time, and they're becoming mixed. So there's a device with an AI, device with a receptor diagnosis, and an AI to read the receptor. So there's no longer a single play in the business. It's really all contributing each other. And although it happened in COVID when we used computer systems and AI intelligence to choose our receptor binding capability and which protein sequence, amino acid sequence for protein binding, it was not really highlighted, although it was part of the engagement. So this is very important, and I think the more we push this information to the public, the better we can then push our what's closest to our heart, biotechnology sector engagement. And I'll show you today some examples when the public sector understood what is needed to be done. Actually, a success has happened. And I spent the last 15 years trying to understand why it's becoming difficult to do one thing over the other. So the first question comes is, why do we need to do it? Of course, this is a very important question because all decision makers, they will tell you right off the bat, no country does everything. And so why do we need a Saudi Arabia? We need to push the field of biotechnology. I created this list, and we need to look at it with an eye of criticism. One is jobs. Why we need to create jobs in biotechnology is because we have a big inflow of people in the biotechnology sector. That's the truth. For whatever reason, in our culture, in our society, in our universities, in our systems, the number of people going through science colleges or applied science colleges or pharmaceutical or healthcare provision are a large number. And those number of people will need an exit of some sort that is fulfilling from a career perspective. So job creation becomes relevant, not because, we don't have, it's a new opportunity. No, we have an ongoing manpower creation. Number two is an income. The amount of investments that has gone through biotechnology in the past 5 years is so significant that it's compatible or comparable to what went into EV, electric vehicles, what went into energy, what went into green energy, what went into hydro energy, what went into green initiatives, what went to space. And so I think it's important to understand that this money is potentially an income. And so the loss of opportunity here is also a big component. Then we talk about healthcare access and cost. There's no country on earth that is not talking about the cost of healthcare services. There's no country that is talking about the cost of healthcare services. But the problem with healthcare services is because healthcare is part of the national economy. If your citizens and residents are healthy, then your economy will be good. And so it's reciprocal. If you maintain the healthiness and wellness of your citizens and residents, your economy will be good. However, you want to reduce the price of healthcare. It's costly, and it's a one-way highway in terms of cash. The finance people see money going. That's it. And so this is why there's no country on earth, and I'm saying in general, that is worried about how much it is going towards health expenditure. You can try to cut costs by doing all the maneuvers that we are doing today. Maximize purchase, improve procurement, reduce waste, increase efficiency, operational efficiency in healthcare. But there is a limit to that. And no matter what you do, you can squeeze it all you want, and you're gonna go over your head 100% to get a 1% cost reduction. The best cost reduction is you invest into the consumables, the downside, the downstream of your healthcare. If you have invested in gloves, then gloves becomes no more a cost, it becomes an economic impact. If you medical gloves, if you invest into pharmaceutical, this becomes part of your local investments. People always complain and say US, for example, has the highest cost of healthcare. They also have the largest economy. The money that is going in there and all the recent Republican move is to try to keep the money inside the country. So the best, most efficient way to reduce cost is you go downstream in the industry. Downstream means all your consumables becomes a local. This will create jobs. This is what the local content government procurement authority is talking about. Now, it's not only about that, it's also access. Access to care. I think the COVID-19 was a good example. Access to certain elements was jeopardized because of all the borders blockage and whatnot. And so I think there is a security aspect to it too. Many countries today, for whatever reason, they got their borders locked down and they have to live with what they have inside their borders. And you need to ask yourself, what can we do if that happens? And I think the healthcare, it's like food and other essential elements that you definitely need to make sure I have it, I can survive. Last but not least is the economy diversification, and I think this is a big discussion as part of the Vision 2030. I think the partner from oil-based revenue is the hallmark of the entire Vision 2030, and so it becomes, okay, among the industries out there, which ones we can do best? And I'm pretty sure this comes on the top of the list, or at least among the list. We look at what is happening from a national perspective, because here the question we have today, what can we do as a country? So let's start with what the country has decided already. Well, the country has decided already, but with few things. The most, I think, known to us and relevant to us was the National Biotechnology Strategy. It came following the National Industrial Strategy, and it came following our National Investment Strategy, and they both were actually, all three were aligned on the final picture that we need to have a capacity, local capacity in vaccinations, manufacturing, genomic testings, and others. So I think the wording that came out of the strategy was very clear that this is among the priorities. The country could have decided we don't need a strategy. We have other things. We have many other things. But if you look at they're all back-to-back تقريبا within 12 months or so. We had the National Industrial Strategy, we had the National Investment Strategy, and then the National Biotech Strategy. And you can see they're all aligned together. And of course, Ministry of Health was play in all these wording, major play in all this wording. And if you actually go and start reading the content of those strategies, this is the copy-paste. I should put quotations, I should not really copyright. So this is exactly copy-paste from the strategy. This is develop end-to-end vaccine manufacturing capability, ensure self-sufficiency, and enable exports across the nation. This is a big mandate. And I can tell you we're at least 10-15 years far from this. Simultaneously spearhead research and development. This is a big component in innovative technologies that become a regional hub, late-stage development. But this is very important, and we're going to come back to this again. The strategy talk about late-stage development, and so this comes from the national strategy. Right, let's look at the ecosystem, and I put it in a way that non-biotechnology people see it. So we, as I was saying, it's very important for us to start to educate the overall public. So we need to talk their language, not our language. Our language is good because we always speak our language between ourselves. Now we need to talk to other people outside the field. And so in the healthcare business, we see four things. We see people, patients, general public, citizens, visitors, tourists. They're all people. Now we have big tourism components. These are people. Then we have data, because everything, all these individuals have data, whether it's data coming from their phenotype, genotype, clinical, non-clinical, these are data. These are all data, right? Then we have things. So things, everything we expect in the healthcare, and we want to, this is our biotech side, but these are things in terms of finance. So these are the products, these are the vaccines, these are the therapeutics, these are the diagnostics, these are the medical devices. These are things, right? These are including the providers, the hospitals, the beds, the ventilators, the machines, right? The scrubs, the cleaners of the labs, sterilization machines. Right? These are all things. You buy them, you have them. And then the process, because at the end of the day, everybody worry about how we are delivering care. So when people come, they bring their data, you apply things, and you do this in the process. Called clinical guidelines, we call it best practice, we call it evidence-based medicine. It's a process. It's a process. If you go to a supermarket, buy something, it is a process. Right? So everybody sees it as a process. And that process includes all the flows, all the records, all the hospital managements, facility managements, the manufacturing management facility systems, and so on. And you need to think, where do you want to play a role? Do you want to play a role in improving the wellness of people? And so we need to push on education, get people to be aware. And so in the recent years, we probably have seen, as part of the National Transformation Program, a big push towards healthiness. And we started to see walking campaigns, running campaigns, sports campaigns, improvement in the cities to try to allow people to be up and about. It is a major impact on health, and it's an engagement that was successful. At the previous end, there is no city in the country that doesn't have an initiative that allows people to come out of their homes and try to participate in an activity. And that's part of healthiness. And it's an impactful engagement in health, but it doesn't involve biotechnology, but it's impactful engagement. We look at the data, and we just closed a major workshop a couple of days ago in Riyadh about data, and there's already new players in the data. When I was at the Ministry of Health years ago, it was really the two major players at that time was the big hospitals that had EMR, electronic medical records. And those were very few. And I remember in the early 90s, Cerner was already at King Faisal Specialist Hospital, where everyone else was still paper-based. So they were having data collection. Now we have Lean, Feast, even new names, right? That has large number of data that is being collected, and included we have CHI who's collecting all the insurance-based data and so on. So these are new initiatives that succeeded. Sedaya came on top. There's a data push. There's a digitalization government authority. So you could see things moved from what it was before. A lot of movement happened in that sector, in the data sector. A whole organization came, it was Sedaya. Right? A whole organization showed up that run and secure data, and now they talk about data more from a regulatory point of view, even stronger than SADC. Then the process has been always a local problem, more or less, and so there was no investment in the process. And again, the things were the investments in the things has not changed much. We talk about, I don't want to make you feel bad, but the first biotech workshop strategy was June 2011, Ministry of Health. It was conducted by وزارة الصحة 2011 مع الدكتور محمد خشاب الله يرحمه. So this was the first biotech workshop strategy was conducted, June 2011. Some of you maybe were not in uni at that time. So and it's many years down the road. Now we have many new entities engaged in the biotechnology and biotechnology strategy. However, we're still seeing it moving slow. And I'll give you some benchmarks of how things came after us and moved fast. Right. So let's look at the biotechnology life cycle. I'm trying to simplify it. It's not that simple. You guys know this more than I do. So I will try to simplify it from a process perspective. We have the first one is the discovery stage. So discovery is a stage where it involves identification, new biological process, products. This is mostly research. This is both basic and applied research. And this is where novel application comes. This is not necessarily therapeutic. This is diagnostic. This is part of therapy. This is prognostic. This is part of understanding the human body, right? And I don't think there's a question. This is something usually led by universities, usually led by research and development institutes, run nationally from academic entities. And I lumped الجامعات احترام التعليم. And I know that you're departing that, but in the current situation, but also RDIA I think came with the whole ideation of supporting research and development, plus our usual other institutes who are involved in research and development. That includes Faisal, Keynote, and and I think there is a lot of discovery happening today. And now, if you want to benchmark what we are doing with the regional ecosystem, actually we always win in the metrics. A couple of days ago, we had RDIA presenting our KPIs related to IPs, patents, filing, registration, approval, compared to the G20 countries, and we fall between 13 and 15 in terms of ranking. And so it looks like we have good discovery. Not perfect, but good. And so there's always a place to improve things. But the current activity is actually reasonable. Then we come to the next phase, which is the development stage. In the development stage, this is where you take promising discoveries, and not all discoveries are promising. So you take the promising discoveries, and this is a very rigorous testing optimization process, so that product can be closer to commercialization. It's not commercial yet, close to commercialization. And as you could imagine, because it affects the human, the development stage is a long stage, is a rigorous stage. I don't want to use the word complicated. It's not complicated, it's rigorous. It's rigorous because it's the human health. This is not something that you could say, okay, it didn't work, let's move it down. This is something that needs to always work, and when it doesn't work, it needs to be proven within a very controlled setup so it doesn't harm the individual. Nobody wants to be harmed. So the development is rigorous, and we have in the country, and our regulatory body, which is Saudi FDA, has been aiming to always reach to the top of the regulation strategy rather than the bottom of the regulation strategy. And so it's really, as part of their strategy, they're trying to be always matching the most stringent regulatory body. Now, this raises the bar for development, but it also gives you a big market entry capacity, right? So you need to see the other side of the things. There are many regional regulatory bodies around Saudi Arabia. If they gave you the approval, it means zip to any country outside your country. So it has no market value. There's no commercial value. Saudi FDA gives you 40-something countries approved, whether directly or indirectly, and that number is going up after they hit their fourth categorization. WHO as a regulatory body, it's opening more and more venues, which is good for the investor, because all they want to know, how much market share do we have? And Saudi Arabia on its own is not a large market share. It's a good market share, but it's not a large market share. So they want to know that if you get Saudi Arabia approval, it means you can hit larger markets. But it makes the development stage more rigorous. And in the development, there's actually way more players, right? And that includes the Zara, that includes hospitals, whether it's private or public. That includes the Saudi NIH, with its new, its published intention, they want to play a big role in the development stage, and that includes the clinical development. We have the RDIA also as part of the ecosystem, and we have the incubator excellence. And I want you to think a little bit in technology, IT technology. Before, in 2018, we knew very little IT technology. We had very little apps in our phones. Maybe Uber, you flew outside the country, maybe Amazon, maybe Expedia, they're traveling. If you look today at your app, there's way more apps that are related to technology. Many have actually came out of the Saudi, and that includes or the region, it's GCC region, that includes Jazz, that includes Kareem, that includes others. And so, if you think about it, how did we move that very fast? And biotech capital, These guys came after, and they're already a unicorn who are billion dollar and above value. How did they do this? And if you think about it, it's because people understood the journey. The public understood the journey. It's very easy. And so if you come to the development stage, the discovery was already happening. If you come to the development stage and actually now look inside the country of the number of incubator accelerators in that business, I don't think you can actually get them all in one search. Yes, there's so many, you cannot get them all in one search. Public and private. And I just tried to pull a few inner 500 Blossom, Impact 36, Wired, Garage, and others. So there's so many incubator accelerators that has supported the development of products. And here, the incubator accelerator play a role, the angel investors play a role. You need pre-seed funding. The pre-seed funding comes usually either from a public sector or from a private sector. The private sector usually are family and friends who are willing to get in. They believe in you and they're willing to give you some of their top of their saving, or angel investors who support you. And if you look at the success of the technology, is because they succeeded in this. They were successful in this. Now, yes, the development cycle is shorter. Yes, the money needed is smaller. Yes, the commercialization is easier, but they understood it, right? This is the same as when we understood how we get into a real estate business, how we get into entertainment business, how we get into hospitality business, right? It's an easier cycle. However, it's technology. This was technology, and so it was a question of, is it going to fail? And so if you today, although we're scientists, and if someone comes to us with an app, you really don't have the capability to do a due diligence to know if the coding is right or not. You could try it, but you don't really know how to do a due diligence coding. But people invested. Although they couldn't do due diligence on the product, they were able to invest because they understood the development cycle. They got it through, and they were able to execute. In the clinical, we have an engagement of clinical trial units, research organizations on top of the providers. And that ecosystem is honestly yet to be mature enough for the healthcare side. But there are various players already. Comes the next one, which is the deployment stage. The deployment stage is also an investment stage. So we're still not commercial. We want to hit commercial. And I started to see some deployment difficulty in the tech space who are healthcare. The tech space who are healthcare, invested in healthcare, although some of products had hit Saudi FDA regulatory approval, they couldn't get into good deployment. There are diagnostics that came from some universities that got Saudi FDA approval, still didn't go through. We have AI solutions that got through Saudi FDA approval, still didn't get through. And because the deployment stage is a complicated stage too, and it does require, it is the final focus, and it's the commercialization and implementation part. You would love that a large multinational company takes care of this from your hand. But the truth is, more and more as we advance biotechnology in the last few years, biotechs are stuck doing this on their own, not a large multinational engagement. So today, if you go back 10 years and look at the sales of pharmaceutical or medical devices, you're going to see all the common names and easy names. And now if you look at the value and the sales that's coming out, you're going to see new names. These are the biotech companies who are stuck on getting into the commercialization by themselves. And I guess Moderna is a good example. And so the Moderna, until prior to COVID, very few people know them. However, they existed almost 10 years before. And they contacted Saudi in 2012, just to give you any heads up. Saudi, outside the US, 2011. I have the email from them, it was 2011. Where I would have just hoped to put on investment that would have been the right thing to do. But I didn't. So I think the deployment stage is a complicated stage. It's not an easy stage. and today has created a negative feedback on the system that we did not successfully deploy some of our innovative solutions that actually gone through the development, got scientific approval, and couldn't get into a successful commercialization. I think King Faisal has some examples, KMR, KAUS, and other universities all have examples of it did go through, but it didn't hit commercialization. And here we have even bigger players are engaged. We have real investors, seed and Series A investors. So these are serious investors and private equities, venture capitals. We have Musha'at, whom they've done a good job outside biotechnology, but they've done a good job in other sector, honestly. Entertainment, hospitality, health, tech, technology, IT. They've done a very good job. We need an industry, we need the multinationals. I think the multinationals are still are not interested in our market, understandably, because they have the globe to choose from. And when you have the globe to choose from, you become picky naturally, and you want only the best. So we are not in that phase yet. I think the only entity that was able to license to multinationals is probably KAUS. The tech transfer offices in our universities are still passive. They're still, although to the best they could do, the KPI is linked to money. It's not linked to how many did you license out. If this is a KPI, I think life would be different. If we tell them you are a percentage of your how many over our dominator. So our dominator is everything we have patent. And your percentage success is how many of those you licensed out. I think their life would be different. Until that's a KPI, I don't think we will get them excited enough to be in business development mode, not in, I want to work with you mode. And if you, I want to take you back to the IT technology. It's a good example. MICT has done a big rule to really make all these three aligned. If you want to think back على جاهز ولا غيرها من الابس اللي طلعت if you want to go back to these technology entities, if you see their journey from discovery to development, deployment, and all the entities that decided في النص and made it easy for them to succeed, US are respected by MICT. And I think this is an important message. And now we're realizing who were able يقول لك اللي حط في طريقك الاشياء الصح عشان تمشي. هي صح موجوده بس في حد حطها. طبعا كل شيء بإرادة الله سبحانه وتعالى بس there's also a function of insertion. That takes me to let's think of where we are and what we're missing in order to have these right things in front of us. And so we start with the first one, the discovery. Discovery is really not too bad. We have very motivated scientific community. It's actually, by the time they lose their motivation because of wear and tear, we have new ones coming in. So that's a reality. But they understand very well the demand. The demand is, it's everyone coming out from science community, they know exactly what's missing. Yes, they are being trained to be more entrepreneurial, commercial, but not necessarily enough. We do have a lot of labs. We do have good infrastructure. Some needs to be optimized, but we have good infrastructure. There are granting opportunities. A few years ago, this was drying up. Thank God this is coming back to life, which is great. And I don't want to be negative, so I'm putting it as opportunities, which are really things we need to fix. We still have bureaucracy, we still have weak collaborations, everybody wants a win. And so this is 10 teams in الدرجة الثانية من الكورة كلهم بيتعاونوا. It's not going to happen. Only 2 or 3 will make it. So if you don't work together, it's not going to happen. And no one is going to have to qualify للدرجة الأولى. Forget الدرجة الممتازة. So we need to also ease and promote transition from public to private. We still have some remnants of our prior culture, which is Vision 2030 came to change and bring the public and the private closer. We still have some left remnants from that culture. The entrepreneurship mindset, this is yes, we're doing it. We need to even move it forward. Everybody needs to learn entrepreneurship, even if they don't want to do it. So at least they don't block it. And you want them to be at least a non-negative. And so you want to get everybody to understand what are you talking about. We need to generate a system to support the industry scale quality service from universities to industries. And until now, if no university really jumps onto the, I'm willing to do it. I'm willing to do it as part of my job. This is not, you're competing with the globe. So we need to get into the, let's work that, to close that gap together. Focus on the pre-seed funding, I think, and try to generate wins and know that, as I said in the technology, there was quick wins that made things move faster. I'll go to the second one, the developers. The developers are also motivated. They want to support localization. There's a great quality of care. I don't think there's a problem with care in Saudi Arabia. I think we're one of the best healthcare service providers in the region, if not globally. The access is pretty good. We don't have access problems, which some countries do have. We have some clinical trial units in the public sector, not so efficient, but we have them. Saudi NIH establishment came really to push the development. These are all good news. Some of the things we need to work on. We need to work on the cost. This is an expensive country. We are not a cheap country. Saudi Arabia is not a cheap country. And I think we know this now. Everybody living here, seeing how much of their salary is going to disappear by the end of the month, knows this is not a cheap country. And so you need to think, if you want to compete, you need to reduce costs. You need to maximize the investment that's coming from the government, because most of the infrastructure is a government investment. And I think the national RDIA strategy to allow access to the labs was one testament to that. Think a lot, push the clinical players to think more about buy versus develop strategies. This is a very complicated strategy. If you want something, you can buy it. Sure, that's an easy solution. But if you develop it, you have more sustainable supply, and you have an ROI, and you have an economic impact. And I think the local content government procurement authority is trying to push that on the public sector. But honestly, this is a national interest. This is public-private. You can buy something, you can buy the iPhone, or you could work on something that's competitive. Only the presence of an alternative gives you more options, gives you more flexibility, gives you more freedom of choice. Think about the path also to the industry. The industry is important. We need to support the local industry further to come closer to the development, not to wait for final deployment. Our industry today is sitting post-deployment. Class commercial, okay. This is not really what we want. We want to bring them closer. This is a big role on the industry side. We want to improve the public-private participations, and we need to increase our CTUs. I think I'm running out of time, so then I run quicker. We need to think of all the ecosystem, not only us as a university. We need to think of the industry groups, we need to think of the investors, we need to look at the economic drivers. And there's so many players out there that we need to engage them into the decision making. I think we have a very good strategy, national strategy. We have a very clear RDIA North Star. There's a clear ambition, there's clear plans of where we want to head. Becoming a regional hub is a big mandate for the kingdom. So I think the North Star is already set. The question is, are we going to reach there 10 years or 20 years or 30 years? And so if I want to think again of how IT did it and what's missing in biotech, you need to think, and this is my personal opinion, we still don't have a leader in the government sector that is pushing biotechnology forward. We have multiple players. We don't have a leader. And I take you back to the MICT and the technology. MICT decided they want to push that forward. They made it. They did not ask anybody. Even when it came to grant, they made their own grant, TPP. They did not wait for RDA, they did not wait for Saudi NIH, they did not wait for anybody. They made their own grants. They made their own grants, they made their own systems, they made their own entrepreneurs, they got their own incubators, they pushed it. They made it incentive for Munshad and others to make this a priority. They pushed PIF and others to not to compete, but to invest. That made it successful. I think this is a great success. So I think in biotechnology, we're still missing that. This is not, unfortunately, it's not a quick win. Biotechnology is slow, as I said, development, deployment, and commercialization cycle is really a long cycle, so it does take a lot of time. 5-7 is even short, actually. But I made it because the finance people usually lose you at hello if you say anything above 5. So I put 5, then pushed it to 7. But you really need to think of a long, medium to long term. There's no short wins, there's no quick wins in this business. Build on the existing system. I think there's a lot already in place. So we need to build on the existing system. We need to benefit from our competencies and flip that forward. We need to successfully think of, we always bench with the G20 as Saudi Arabia in many things, women empowerment, green initiatives and environmental, energy section, and patents. I think this is the time we also need to bench with G20 in the industry, in the biotech industry, and no longer bench with nearby countries. And I think this is one of the issues that is not being addressed. So when we talk about we talk about instant manufacturing as our biotech, we're not really benchmarking with G20. G20 countries have done this 30 years ago, 3-0, right? So this is not the right benchmark. We need to really think proper benchmarking so that we know where we want to head. And then I think healthy ecosystem is an important ecosystem. We need to be as much as possible collaborative. This is a new initiative. This needs everyone's support. Everyone needs to add something. I think a competitive mindset in here, and what can I do, what they're doing, and even presenting that to third parties is actually weakens us. It doesn't strengthen us. We need to be collaborative and non-competitive. And I think that's all I have to say today. Thank you very much.
Allah يعطيك العافية, Prof. Mazen. It was a great lecture, and I guess there is a lot of questions maybe in the discussion panel, inshallah. Now I will maybe introduce Dr. Ali Hassan. I will let my Dr. Ayman, you can sit. Dr. Ayman to introduce him, but before I know Dr. Ali Hassan for a long time. He's one of the few successful entrepreneurs, and one of the few successful stories in biotechnology with Nanoparticles. I hope that he will maybe give us a little bit about the company. Thank you.
Thanks again, Prof. Mazen, for your excellent presentation. Now we have our next speaker, Dr. Ali Hassan. He is the Associate Professor at King Abdulaziz City for Science and Technology and Adjunct Professor at Al Faisal University. Dr. Hassan was a postdoctoral scholar at the University of California in 2015. And also Dr. Hassan received his PhD in the Department of the Biological Science program from the Northwestern University. So welcome, Dr. Ali.
Assalamu alaikum. Thanks for the great introduction, and thanks for organizers for the great invitation as well. Hopefully, I'll give you a little bit of a story to complement the great keynote talk that we just witnessed. And I'm going to share with you why we want to transform gene therapy. As most of us know, current gene therapies are for the few patients, but not the many. Scalability is a big issue. Why scalability is a big issue? Because of two facts. We have 300 million patients around the globe with 10,000 different genetic diseases. How can we treat it? And when can we treat it? The current therapy is really lengthy. It requires patients to be in and out of the hospital for almost a year. And the cost, probably you witnessed in this era, the most expensive biotech products ever. A drug ranges from $2 million one dose to $4 million. And the problem is that the dose price keeps increasing. And this is honestly because it's being done ex vivo, outside the patient. So patient cells are collected, shipped from Saudi Arabia all the way over to overseas like US, and then in a petri dish, the gene modification, gene editing is happening on patient cells, not the actual patient. So this is a bit increases the cost of logistics, the materials used like viruses, the transplantation cost afterward, like stem cell transplantation and beyond. So we want to change this. We looked at different successful systems that could enable the transition from ex vivo to in vivo. And no surprise here, viruses are the most common. They are really great in hijacking our cells and making genetic changes. But viruses have limited cargo when it comes to gene therapy. They always require a plasmid DNA. Can you insert a small RNA inside them? No. Can you increase the size of the plasmid DNA beyond 10 kb? No. Can you just package them with RNA? Again, no. So we need to think about innovative technologies, something non-viral per se. And to be honest, COVID-19 opened the door for what we call lipid nanoparticles. It was the soldier behind the success of mRNA therapeutics vaccine at that time. So yes, lipid nanoparticles are great, and we think we can start there from COVID and build on it. And then we need to identify what are the challenges with the current lipid nanoparticles, so we can take them to the next level and hopefully become a really transformative carrier for gene therapy. We know at the moment they have low efficiency, they have some potential toxicity. If you look clearly, if you dig deep really into literature, you will see most of our scientists design those lipid nanoparticles and produce unstable structures. They break down easily in shipment, and they break down easily inside our body. So they are also designed with toxic components. When they break down inside our body, the medicine cargo inside them will end up in liver and will die off because it hits its target. And this is, again, has to do with the third biggest challenge, the non-specific delivery. Again, they break down, even if you add your active targeting or passive targeting. If they break down, the medicine will not reach to the right target, right organ or right cells. So what I'm going to show you today, how we collaborate with AI this time, not a human, to produce the product that we want to test, to discover the products that we want to test. And hopefully I will give you a quick example about a few monogenetic diseases like sickle cell disease, retinitis pigmentosa. but hopefully we will continue this journey in the next 10 to 20 years to tackle hundreds of genetic diseases. The common problem that we're trying to solve is stability of lipid nanoparticles. Now, there is a really nice piece of equipment present here in Jeddah ecosystem and in the other ecosystem as well. It's advanced nanotechnology called nanolithography. Today, originally is used to create screens, TV, smartphones, because this is what's designed for, create chips, semiconductors, and so on. So we saw an opportunity. Can we solve the dilemma of lipid nanoparticles? Can we use this piece of equipment to create a stable lipid nanoparticles? And of course we can. The difference between what everyone is doing in the past 30 years working on lipids and what we do is this. Everyone is taking raw phospholipids and then mix them, simply relying on nature to self-assemble. What we did is we took those three raw materials, but then patterned them, create a block. So we changed how precursors are being used, then rely on self-assembly. And what you see in the middle is a chip 2 by 2 centimeters. In 5 minutes, you can go to the lab, print out millions of lipid nanoparticles with high stability and high size, uniformity, and morphology as well. Everyone working on the lipid nanoparticle knows it's not easy to defeat and create nanospherical. Most of our lipids are spherical, but with this method, patterning, you can create cubics, you can create rings, you can create rods, you can create cell-like structure, exosome-like structure, you name it. But again, this is now trained in AI. Now the AI can spit out the recipe. The AI can read your microscopic images, analyze those nanostructures, extract features from you. Now the AI can look at single lipid nanoparticle from 100 different dimensions as opposed to humans. And it will ensure so many criteria in your design. Stability, safety, so it has to be designed by safe components. And of course, it can tell you how to target lung, how to target specific cells like rods, and so on. And this is really interesting because when you image them, you can feed them in this algorithm, and it will start to detect every single lipid nanoparticle, and it will check their morphology, their sizes, and their chemical composition. Accelerate our R&D from 4 to 5 years to a couple months. So this is an amazing tool to collaborate with. And this is what's inside this algorithm. You can play with so many experimental parameters. The AI will generate those parameters for you, and you will play with 56 different outputs and results. I want my efficacy to be this level. I want my toxicity to be this low level. I want my disease and organ is this target, and so on. So it will spit out based on your inputs. You can talk to it, and you will get results like this. The energy, the surface roughness, the composition, the sizes, the morphology, everything you could dream of, or everything you could do that will require a couple of years to do. So we're happy that we have really interesting structures. Some of them does not exist in nature, actually. but some of them are reminds you of some natural products like corals, like cells, like capsules, peanut butter maybe, and so on. So you can really have fun with it. We have 800,000 different structures. Those are just a quick example. And my favorite is the capped exosomes at the bottom. Now we can do injectable. Now we can change the game here and go from ex vivo to in vivo. When you do that, you would expect the cells to go bind to the diseased cells, snip the DNA, erase the genetic mutations. We have tested this in so many cell lines in animal and also in human as well. You can, in less than half an hour, you can see them inside the cytoplasm, high transfection efficiency that I think is one of a kind, while minimizing cell toxicity as well. We introduced them to animal IV or tail vein injection in this case, and we expect them to escape liver and go to a certain organ, like lung in this case. And this happens in not only quick, like 2 hours, but it stays for a couple of days. This is just the 2 hours biodistribution. And for the record, the level of lung to liver is huge, 6-fold. Most of the targeting, you get 50% in liver, 50% in your organ if you're happy. But this is a huge, like 90% in the lung, really minimum in the liver. So this is the same message. Also, we challenged the algorithm. Can we go for specific cells as opposed to specific organ? We have a disease called retinitis pigmentosa. Usually at the current, Novartis is designing viral to be injected in the subretina and involves long hours of surgery. What we wanted to do is to become non-viral and to go to specific cells like rods within the retina. Those working on lipid nanoparticles, no, we cannot cross the retinal layer with lipids. No one was successful, but luckily with this steel structure, you can see the morphology, it can go all the way from the intravitreous to rods. I want to speed up to this important slide that reflects the ecosystem. We couldn't have done it without the infrastructure, like CACS and the National Guard Hospital. MCIT was really instrumental in funding us. We raised almost $2 million just to do this job. The role of incubators, as Dr. Mazen mentioned, like El Garage and Neom Autonomous, were really instrumental in shaping this. This is a journey we started back in 2022. Started as a spin-off from CACST. We received grants from the MCIT, incubated by multiple incubators. The National Guard Hospital in Riyadh, in our backyard, was really willing to try our product on patients, on a human. So you can see the ecosystem is happening. This is an example. Hopefully everyone can be optimistic as well. So with that, I want to end here and thank you.
Thanks so much, Dr. Ali, for your free talk. Now, let's move now to the next speaker. Please welcome Dr. Hussam Al-Tayyib. Dr. Hussam Al-Tayyib is the Director of the Center of Innovation in Personalized Medicine at King Abdulaziz University. He also served as an Associate Professor of Nanomedicine and Biotechnology and the Faculty of Applied Medical Sciences, a leading researcher in the field of nanomedicine. Dr. Tayyib work focus especially on the development of personalized nanotherapeutic and nanovaccines, aiming to revolutionize the treatment and prevention of various diseases. Dr. Tayyib is an accomplished inventor holding 3 patents from the US Patent and Trademark Office. These patents focus on the innovative nanoemulsion-based cosmeceutical and 3D printed nano-encapsulation designed for advanced drug delivery and personalized vaccine development. These inventions showcase his expertise in translating cutting-edge nanotechnologies into practical application with the potential to significantly evolve healthcare outcomes. So welcome, please, Dr. Tayyib.
Assalamu alaikum wa rahmatullahi wa barakatuh. I think Prof. Mazen and Dr. Ali is, for me, the top to start with. And Dr. Ali talked about lipid nanoparticles. And I'm going to talk specifically today about one class of lipid nanoparticles, which is nanoemulsions or nanoparticle emulsions. So the pharmaceutical industry in Saudi Arabia is considered the largest in the region, and it's emerging as a promising destination for investments. And that's why many companies, or multinational, multi-international companies, come into Saudi Arabia to invest in the pharmaceutical industry. However, the Saudi pharmaceutical industry compared to the global industry is having limited R&D. Also, we are only dependent, or mainly dependent on generics as well as imports from outside. So we should focus on developing new pharmaceuticals based on compounds or novel compounds that have promising therapeutic properties. However, the therapeutic candidates are limited by many challenges, and they fail to reach the pharmaceutical market. And it's estimated that the success rate for novel compounds to reach the pharmaceutical market is about 5%. So what are these challenges? I think Prof. Mazen talked about the regulatory challenges. There are also physical and biological challenges, and these include poor water solubility, degradation, stability, also biological barriers when we apply these drugs or these candidates to the human body. So nanotechnology came here to help these therapeutic candidates that might fail to reach the market to succeed through different mechanisms. One of them is encapsulation. So encapsulation can protect these therapeutic candidates from degradation through, for example, light or oxidation, or even inside the human body by immune responses such as protein corona or macrophages. Nanotechnology also can protect the human body from the side effects comes out from these therapeutic candidates. So nanotechnology has many applications, including pharmaceutical applications. The medical applications of nanotechnology include diagnostics, medical imaging, therapeutics, and vaccines. So what is the nanotechnology? It's the development or the assembly of nanoscale particles from 1 to 999 nanometer. Nanotechnology are available in nature, so many nanomaterials are available in nature that we don't know about, but it's discovered. And one of them is the nanofibers that are found in spider cells. Also, ocean sprays, volcanic eruptions also synthesize some nanoparticles that are toxic to us. So how nanoparticles, or specifically nanoemulsions, are fabricated? So there are many different ways, but they depend on energy. And for example, here we have the ultrasonication method, which we break down larger macro oil droplets into nano droplets. And this can be done through ultrasonication or high pressure homogenizers. And then after we formulate these nanoparticles, we can build up the surface of these nanoparticles to suit certain medical applications. And I just want to stress here, as Dr. Ali said, that most of the generic surfactants or the stabilizers, they don't allow for custom functionalization of these surfaces. And we're gonna show how this can happen using certain specific technologies. So nanomaterials are classified based on different classifications, some of them based on rigidity, materials, chemical nature. And there are many different types of nanoparticles. Lipid-based nanoparticles, I think, the most promising in the medical field, and they have made great success in different applications. The most maybe common ones are Pfizer and Moderna vaccines, and they are lipid nanoparticles encapsulating messenger RNA that were applied for vaccinating us against COVID-19. Doxil was the first lipid nanoparticle that was approved in 1995 for the treatment of cancer. So to develop nanoparticles, we need to overcome certain challenges through different administration routes, and this includes, for example, the skin route. Many of the active compounds or the therapeutics cannot penetrate the skin. So using nanoparticles or using nanoemulsions, we can penetrate the skin to deliver these bioactive compounds to certain skin layers. As Dr. Ali also said, there are different ways to target these nanoparticles. One of them is passive targeting, and the other one is active targeting, which is labeling the surface of these nanomaterials to target certain receptors on the surface of cells. So the lipid nanoparticles that are generated in the literature mostly suffer from functionalization of the surface, because most of the surfactants available commercially, they cannot be changed, they cannot be functionalized with antibodies, with antigens, with different biomolecules. And actually here we want to talk about how we tune the surface of these nanomaterials to suit certain biological application. So what are the current research at the Nanomedicine Unit at CIPM? Basically today I'm going to talk about nanoemulsions. So there are different classes of nanoemulsions, are single nanoemulsions and double nanoemulsions. Single nanoemulsions can encapsulate only one kind of drugs or molecules, either hydrophilic or hydrophobic. The double nanoemulsions can encapsulate double compounds of two different chemical natures. Lipid nanomaterials, as I said, they succeeded to reach the market through, for example, Diprivan. Diprivan is anesthetic formulation made using nanoemulsion, and it's currently used in operation rooms for many operations. There are also other products made through nanoemulsion technology, but most of these products are not specific or not targeted. So about safety. So when we talk about safety, nanoemulsions are being used for a long time in the medical field as well as in other applications, including food. So butter and milk and ice creams, all of these are based on emulsions. So we've been consuming emulsions for a long time. So what are the definition of emulsions? Are just a mixture of oil and water stabilized by certain biomolecules, including proteins, lipids, and other molecules such as carbohydrates and polymers. They range in size from 20 to 500 nanometers. They're kinetically stable and non-equilibrium systems. So I will talk now about our research. So Salman is my student. He was aiming to target colon cancer cells, specifically transferrin receptors. So here we developed, using our in-house peptides, the system that can target transferrin, and then transferrin can be masking the nanoparticles that will target HCT cells or colon cancer cells that have overexpressed the transferrin. So as you can see here, in the green color, we have targeted the cells, although there are some non-specific interactions. Here we have also developed nanoemulsions that can be co-loaded with two hydrophobic compounds, vitamin D and frankincense oil, for the treatment of osteosarcoma. And we have killed about 80% of these cells at a very low concentration of the encapsulating compound, about 0.02% of the frankincense oil. Ghali, also master student, she helped us to develop a vitamin D loaded nanoemulsion as differentiation promoters for the treatment of leukemia. As you know, leukemic cells, there are leukemic patients, they have many blast cells that are not functional, and they need to be transformed into functional cells to improve the healthcare outcome of leukemic patients. And we have also here confirmed that we can target these cells in the red color there, and we showed very significant targeting compared to non-targeted nanoemulsions. So here also we used the in-house peptides that can absorb on the surface of nanoemulsions to label the nanoparticles or the nanoemulsion with polymers and DNA for vaccine development. And as you can see in the far left of this slide, that once we add the DNA, the zeta potential or the surface charge of these nanoparticles change from positively charged to negatively charged, which means that the DNA absorbed on the surface of the nanoemulsion. Here again, we showed that we can play with the surface of these nanoemulsions, so we can gradually increase or decrease the surface charge of these nanoemulsions in biological applications. And I think, as everyone knows in the nanomedicine, the surface charge plays a major role in the interaction with biological cells, and also in avoiding non-specific interactions with cells within the human body. Also, we showed there that when we play with the surface characteristics of nanoemulsions, we can control the release of the encapsulated drugs. So if we increase the density of the polymers attached on the surface, we can minimize the release of the loaded drug. However, when we reduce the density of these polymers, we can see a huge release in these formulations. And this shows that we can tune the release of these nanoparticles based on the biological application that we are aiming for. This is also just a way of showing how we can reduce non-specific interaction. You can see the B100L, which is the linear polymers on the surface of these nanoemulsions, have higher non-specific interaction to cells compared to the armed polymers. The armed polymers, it's brushed polymers with the same mass, same molecular weight, however, with much lower non-specific binding. So we need to avoid non-specific binding and enhance or increase targeted delivery to enhance the therapeutic efficacy of the therapeutic candidates. We showed there that when we absorb the targeting molecule on the surface, we have seen that the encapsulated compounds, which was fluorescently labeled, was internalized within the cells using the targeting molecules. So antimicrobial resistance is another major health problem that we were able to target. And here's just a simple way to show how nanoemulsion can be co-loaded with different molecules. So we co-loaded the nanoemulsion with antibiotic plus an essential oil, and we have reduced the minimum inhibitory concentration of the meropenem by 16-fold. And this is through passive diffusion to the Klebsiella neonate cells. We have also formulated a nanoemulsion loaded with Saudi origin plants called Artemisia absinthium, and we enhanced their antimicrobial activity by 32 folds. And most of these emulsions, they are stable at least 6 months in the fridge. So we've tested them in different biological materials, and they show stability. I think I have a limited time here. So we've also developed another formulation against Staphylococcus aureus, and they also show promising antimicrobial properties against Staph aureus, either in MSSA or in MRSA. And finally, we have developed multiple patents using these nanoemulsion compositions, and one of their properties is that they last for at least 1 year in the fridge without any change in the PDI or in the size or in the overall physicochemical properties. So we have also explored solid nanoparticles, but for different applications. Here we explored it in forensic applications. And I'm not going to talk into detail in this. And finally, so the share of nanomaterials in the pharmaceutical market is rapidly growing. Lipid and soft nanomaterials are safe and offer huge opportunities to translate therapeutic candidates into tangible healthcare products. Nanoemulsion can enhance therapeutic properties of many compounds. Antimicrobial nanoemulsion can offer new solutions or new antibiotics or next generation antibiotics. Nanoemulsion can serve as vaccine platforms for future superbug threats. And regulatory bodies are given larger attention to the regulation of nano-based drugs. Finally, I would like to thank the team at the Nanomedicine Unit and at CIBM for their contribution.
Thank you, Dr. Hassan, for your nice presentation. Now I will give the mic to Dr. Rabir to introduce it.
The next speaker is Dr. Mohammed Al-Faleh. He is a biotechnologist with a PhD from University of Queensland. specializing in biotechnology, biologics, and he previously served as the Associate Professor of Biotechnology at King Abdulaziz University, and also worked at Vaccine and Immunotherapy Unit here in King Fahd Research Center. Dr. Al-Falih also directed the biomanufacturing training program, which is a collaboration between Saudi Bank and King Abdulaziz University and Sartorius company, a global leader in bioprocessing technology. Now يعني بعد ما ختم الدكتور محمد الفالح everything in academia, he moved to the government, in Ministry of Industry, to help building the ecosystem in the kingdom, joining the Ministry of Industry and Mineral Resources as a biotechnology expert. I guess you are gonna hear a lot of interesting things from Dr. Al-Falih.
السلام عليكم جميعا. It's good to be back. اليوم البرزنتيشن حيكون speaking Arabic English. البرزنتيشن اليوم حيكون mainly scientific, related للفترة اللي كنت فيها still فيها بالvaccine immunotherapy unit. Second half of my participation حتكون wearing a hat in Ministry of Industry. talk اليوم حيكون عن engineering antibody like molecules for COVID treatment and prevention. كيف ادي presentations. في البداية الانتيبوديز كلنا نعرف monoclonal antibodies B shape, large biomolecules 150 kilodalton. Antibodies have been engineered, produced through biotechnology means for treatment of different kinds of diseases. It has high affinity to different epitope antigens and polymers. Antibodies is not new technologies. It's over 120 years. During the last 120 years, antibody-related R&D activities have been subject to 4 Nobel Prize. The trend of the antibody market is still exploding. Now 40% of the pharma pharma industry is biopharma, biotechnology related product. 80% of these biopharma is monoclonal antibodies. They represent more than 50% of the top 10 selling drugs. And growth still coming up, especially after the latest advancement in protein engineering, AI, bioprocesses, development, chemical engineering, producing different modalities of next generation monoclonal antibodies like the antibody drug conjugate, multispecific antibodies. So these different engineering approach gave the antibodies new functionalities to treat different kinds of diseases like solid tumors, cancer, inflammatory diseases, chronic diseases. The antibody, the FC region, has the FC region and the Fab region. Fab region bind to different epitope. The FC region is so important that it gives the immune stimulating effect. It also helps with biomanufacturing because we do the purification through the protein A, which has high affinity to the FC region. The FC region has been subject to different engineering approach, different mutations that add new functionality to the FC region, extend the half-life, stimulate the immune system, or can be engineered to conjugate different molecules. Different type of engineering the FC to increase the half-life of the antibodies is through enhance the binding to the FC neonatal receptor, which is responsible to increase the half-life. But some mutations enhance the binding to the FC neonatal receptor, but doesn't extend the half-life. But it can be used to in autoimmune diseases to reduce the innate IgG high titer. or to engineer the FC region to enhance the immunological reaction like the ADCC or CDC or the ADPC. Also, the FC allow the conjugation of different kinds of molecules like cytokines, proteins, that give the antibodies different functionality and different therapeutic options. And this is an example of some of the approved FC fusion proteins in the market or in late clinical trial. So we inspired our work in the last 3 years from the using FC as a building block to fuse different kinds of proteins. So in this example, in the last 3 years, we worked to engineer the FC as a receptor trap, decoy receptor. So generally, the COVID or MERS viruses enter the cells through definitive gate receptors like SARS-2 enter the cells through the ACE2 receptor. MERS entered the cells through the DBP receptor, so the virus entered the receptors through the receptors, so the cells multiply and start infect other cells. So when you engineer antibodies, monoclonal antibodies against the virus developed mutations, escape mutations, or against neutralizing antibodies after giving the vaccine. So we thought that, and other people did the work before us, that's why we did the literature review about the ACE receptor, and we included our novel DBP for FC receptor, decoy receptor. So the idea is, if the virus start developing mutation, escape mutation against these decoy receptors, the virus will become non-infectious. So there is no harm if because we want this virus to develop these mutations, so it become less infected. So we engineered different decoy receptor like the ACE2FC, DPP4, and from the literature we found a very effective modified ACE receptor that we include in our study. And to do the comparison, we included 2 monoclonal antibodies, AB1 against SARS-2 and M336 against MERS. We express these as a recombinant proteins through shell cells. We provide them using a protein A column. We convert that through SDS page and ELISA. Then we engineered pseudoviruses to test these molecules. The pseudoviruses that we engineered, for example, for the SARS-2, we engineered different pseudoviruses based on the mutations of the known serotypes of the receptors, like alpha, beta, and omicron. And we compared all these molecules. What we found that MERS, SARS-2, induced mutations that escaped the antibodies, but no mutations was able to escape the decoy receptor. And this behavior was consistent through the different serotype receptors. And thus confirmed that these decoy receptors can be employed as a fast approach to against epidemic diseases, virus epidemic diseases, outbreak, sorry. And against MERS, we developed pseudoviruses based on mutations that in camels and human. And these mutations, we focus on mutations that in the receptor contact antigen or the antibody epitope to know if these mutations can affect the binding of these decoy receptors. Interestingly, different than SARS-2, MERS antibody against MERS M336 was able to inhibit all the muted serotypes. But it lost a lot of its affinity, although M336 is so strong, in different mutations like B534L. It lost 46-fold from its original inhibition IC50. The DPP receptors was able to inhibit, but it also lost some of its IC50 power with some mutations. Maybe this explains why MERS was less infectious than SARS-2, because these mutations didn't increase the binding or sustain the binding affinity to the gateway receptor. In another part of our study, we said, okay, we want to use this FC engineering technique not to treat but to prevent. So we've used a new approach is FC-based vaccines. So they've used the antigen of the virus with the FC to stimulate the immune system, taking the advantage of enhanced pharmacokinetic of the FC, the half-life by binding to FC nanoparticle receptor. We developed 3 versions. We said, okay, we want to use this FC to protect, to be like comprehensive vaccine, broad vaccine that can inhibit MERS and SARS-2. So we've used the RBD of MERS and SARS-2 to the FC receptor. And we use different format. From the C-terminal, we've used the MERS, and N-terminal, we fused the SARS-2. And then we also developed a reverse version of that to understand the impact of the hinge region, which has a role in the immune activity, ADCC. And lastly, we developed a vaccine with engineering the FC receptor to increase the binding to the neonatal receptor, which is known that highly expressed with antigen-presenting cells, to know if we kind of localize the vaccine within the antigen-presenting cells, do we get more immunity or not? And this is the mutations that we include. It's a property mutations by Argenx, a biotech company that have this drug. We've got it used in mesenteria graphs because these mutations allow to the binding to the FC nanoparticle receptors at in a pH-independent matters. So these neonatal receptors, when they occupy the these antibodies, when you occupy the neonatal receptors, it will not allow the inherent IgG to bind to it, so the inherent IgG will be destroyed through the exosome degradation. So by doing that, we reduce the IgG antibodies within the body and reduce the autoimmune disease indirectly. So we produce these different vaccines using ex vivo system purification using protein A, and we get a good amount of the protein, 5 mg per construct. And then we did STS and ELISA to confirm, STS to confirm the expression and the purification of these different vaccines. Then we did the ELISA to measure the IgG titer for this vaccine. For the Bivax native, wherein the MERS close to the hinge region and SARS-2 at the bottom, the vaccine produced strong immune titer against MERS, but very weak against SARS-2 and SARS-1, even though SARS-1 we didn't include. And when we compare that, I think this explains that MERS has more immunogenicity and might mask the presence of SARS-2. And when measured the reverse, wherein the SARS-2 close to the hinge region, the vaccine didn't work very well, although it did produce some IgG neutralizing antibody against MERS. And lastly, when we mutated the FC region to bind to the FcRn receptor, we got much better antibody titer against MERS and SARS-1 and weak against SARS-2. We still don't know why is that, but maybe there is like some shared epitope between SARS-2 and SARS-1, which is like when we did the alignment was 70%. And lastly, when we did the neutralization assay, the antibodies that has the mutation again with the high binding with the FcRn receptor, it was the best candidate, and surprisingly, it produced antibodies, strong antibody titers against SARS-1, although it didn't include, and weak against SARS-2. And I think we need to do more research to understand why is that, and understand maybe from antibody engineering concept, and maybe change the linker or enhance the structure of the RPD could be the reason. Lastly, antibodies is versatile molecules. It's like a building block. You can fuse different molecules to it to get better functionality or new functionality. The biomanufacturing aspect is well known, well established. So it's like a fast approach to produce Fc-based vaccine and decoy receptors or monoclonal antibody. Lastly, I would like to thank all my students, friends who work with me in this research, and Dr. Anwar for the support he provided. Thank you.
Thank you, Dr. Mohammed, for your public presentation. Our final speaker in this session is going to be Dr. Mohammed Al-Masgalebi. Dr. Mohammed received his BSc degree in animal biology from the University of Mohammed in Morocco. He moved to France, where he obtained an MSc and PhD in biochemistry from the University of Montpellier with distinction. He spent 6 years as a postdoctoral research associate in Imperial College London in the UK, and 1 year in the University of Pennsylvania in the States. Dr. Mohammed received a prestige fellowship from the Wellcome Trust in Open competition. He spent 13 years as a lecturer in protein science in the Department of Biochemistry at the University of Leicester, UK. He is currently a lecturer in Aston University in Birmingham and a visiting lecturer in King College London. Dr. Mohammed has published over than 40 articles and supervised more than 9 PhD students. He has recently been awarded a fellowship of the Higher Education Academy in the United Kingdom. So please welcome Dr. Mohammed.
Okay, so thank you for your very kind introduction. Of course, I'd like to thank the organizer for the invitation. It's both an honor and a privilege to be here with you. And so, ladies and gentlemen, assalamu alaikum wa rahmatullahi wa barakatuh. So in this, the next 15 minutes or so, I'm going to try to talk about 2 things, if possible. In the first 7-8 minutes, I'm going to give you a flavor on my research and interests over the last 15 to 20 years, so just a little bit of a flavor of my research interests. And if I have time, I will tell you a little bit about a story, a small story that's published last year, which is identifying some novel basically compounds that have potential therapeutics for subcutaneous diseases. So what's the biological question? Well, we work on muscle, and of course, muscle is a very important issue. You're breathing, that's because of muscle. If you talk, because of muscle. Your eyes work with muscle, and so on. But of course, that's the muscle which is associated with the skeleton. But muscle, of course, there are other tissues like the vital organ, which is the heart. And the heart, of course, is a muscle, is a pump, but the pumping action relies on muscle. And of course, muscle exists also in various internal organs, the so-called hollow organs, like blood vessels, for example, like the uterus, the bladder, and the lungs, and so on. So all of those tissues have muscle, and muscle play a vital function in that. Now, muscle is one of the most intensively studied systems for the last really 60, 70 years. And so the main mechanism already established by the 60s, 70s, and so it's in the 60s, 70s of the last centuries, they showed that the unit that work in muscle is the sarcomere, and I'm sure you've done this probably in your biochemistry physiology lectures. And this sarcomere is made of 2 proteins, or 2 filaments made of proteins. The myosin filament, which you can see in green, and the actin filament, which you can see in orange. Now, in the 80s, or a little bit earlier, in the 70s maybe, it has been shown that the muscle contract by this so-called sliding filament theory. Okay, so the 2 filaments, they slide between each other, and that's what drives contraction. And then in the 80s to early 90s, so the sliding filament has been shown to be caused by the myosin head, so this protein that basically acts as a rowing protein. So basically they bind to the actin filament and then they pull. And in the 90s and 2000s, and this is the work really of probably over 100 research groups worldwide, a lot of them in the US and in the UK. In the 90s and 2000s, really detailed mechanism up to literally atomic level. So today we have pretty much a very detailed and minute understanding of the mechanism of how basically what you see at the macromolecular, at the cellular, at the tissue level, we can explain it from the atomic level. So now, very briefly, I actually only use actin and myosin as tools. What I work on is on the regulatory process, and on the regulatory process, we don't yet have a mechanism at this level. And understanding the mechanism of these systems is incredibly important because for two reasons. The first one, if you want to understand how pathologies work, how mutations work, then you need to have a mechanism of how the normal stuff work. And the second thing, of course, if you want to design some drugs in a very rational way, then you need also to understand the mechanism of these proteins. So I work on cardiac muscle, and cardiac muscle work nearly exactly the same at the molecular level. But there are some small differences, and that's what I want to focus here on. So in skeletal muscle, if you want to generate more force, you recruit more and more fibers. So there are a lot of fibers in muscle, and so you just recruit more and more fibers. In the heart, that system does not work. In every beat, every time your heart beats, all the cardiomyocytes, all the muscle cells in the heart are recruited. But of course, that gives you a problem because we know that, of course, cardiac output increase between if you are sleeping or if you are in intensive exercise. So cardiac output can increase by as much as 500%. And so if you can't increase, of course, the number of cardiomyocytes because they are recruited in every single beat, in the so-called functioning system, what does that tell you? It tells you that the increase in cardiac output is inside the cardiomyocytes. So there are systems inside the cardiomyocytes that allow you to generate more force. And so this system basically is simply recruiting more and more myosin molecules. So, and that is done through calcium. So calcium not only is a switch, because of course muscle need to relax, so it goes on and off. So calcium acts as a switch, just like in skeletal muscle. But in cardiac muscle, it gives you a little bit more than a switch. It allows you to recruit whatever number of myosin is required in a particular beat. And this is an incredibly refined system to the point that if the system is not accurate, within minutes we can develop edema in our labs. So it's an incredibly sophisticated system, really to tiny amounts. And so normally what I show you here in the curve that the calcium sensitivity, so you can goes from completely inhibited to completely activated. In normal situation, you activate about 40% of these myosin molecules. This activation is affected by both physiological situations, but also by pathological situations. So physiological situations, for example, if you're running, so that the blue curve, so then the calcium sensitivity curve is shifted to the left. And in pathophysiological situations, like for example in heart failure or in various cardiomyopathies, which I'm going to talk about, it's actually diminished, so it becomes compromised, if you like. And so the system that allows that is shown here. So these are the proteins that are responsible for this regulation and for this fine tuning. So that's the biological question. Now the reference question. You have probably heard a number of very healthy athletes, and they are very fine, they are perfectly healthy, and then suddenly they collapse in a sport ground, and they die within 1 hour. And so this is one of the symptoms of the so-called genetic cardiomyopathies, and of course it's a very harsh symptoms. If the first symptom is sudden death, then it's a very serious one. Okay. And so this has, of course, attracted a lot of attention. So in the 1990s, one of the group in Harvard identified the first gene. I'm not going to go through the details of that. But since then, I'm going to just say these carbohydrates are characterized by two things, by phenotypic variations. So there are a lot of different phenotypes. I'm showing you here two. So on the left, you can see a very enlarged, a very thick heart. So that's hypertrophic cardiomyopathy. In the middle one is the normal heart, and to the right you have a dilated cardiomyopathy. And even in this hypertrophy that I'm showing you here, you can have variations. You can have the one like you see here. You can have some just in the apex, in a particular part of the heart, and so on. So there is huge phenotypic variation. And there is a huge genetic variation. Back from the group that I said from Harvard identified the first mutation in the myosin sometimes around 1994, I think. Today, over 2,000 mutations in over 20 genes have been associated with these diseases. Okay, so over 2,000 mutations. About 70% of these mutations or so are genes, the myosin gene, myosin binding protein C gene, and the troponin gene. Now, with all due respect to geneticists, of course, they can sequence anything, they sequence a lot of stuff. Maybe in 2 weeks, they identify a lot of mutations, and they leave us with a big problem. Because we have to do the studies, and these studies, they take months or years to actually try to understand the impact of this mutation. Of course, understanding the impact of a mutation will involve a multidisciplinary approach. So you can do studies, of course, on humans, like clinical studies, epidemiological studies, the omics, the cell studies, the animal model studies. We do none of that. What we do molecular studies. And so we do a lot of cloning, mutagenesis, protein expression, protein purification. Then once we obtain our pure proteins and mutants, we then do the characterization by various biochemical and biophysical methods. Some of the methods are described here. So we use kinetics, and so a lot of kinetics to describe the changes in the pathway, in the biochemical pathway, or we use structural methods like NMR, and I'm shown here, so nuclear magnetic resonance spectroscopy, to identify minute changes in the structure that might be induced by these mutations. And we also use some functional assays, okay? So when we purify these mutants or these proteins, we do functional assays. So one of them is shown here. So this is called an in vitro motility. But you can see the white little things or actin filaments that has been labeled with a fluorophore. You put them on a bed of lysine, and so they are static. And then you add ATP, you see them moving in there. So it's basically a functional assay that basically shows you motility of this system. And then, of course, we can do a lot of measurements, and we can measure the fraction motile, and we can do a lot of variations like mutations and calcium and so on. So one of the first things we have done, for example, for these mutants, and then, as I said, I'm going to show you briefly some of the data. We've used cryo-electron microscopy and 3D reconstruction, so you can see the electron microscope of the filaments to the right. So if you do a lot of averaging has one sequence, it reduces errors. And then, of course, you do some image analysis, and you can improve the quality of the image. And by improving the quality of the image, you can see the details of these actin filaments to the left. And then you have the film filaments minus calcium plus calcium. And what you can see maybe that this strand here in the inhibited state and in the activated state is moving for the white. But for this mutant, what we've seen is that this strand does not move. And so there is a change in the ability of that strand to inhibit the filaments because of that mutation. So I'm going to go a little bit faster. So we've done also like kinetics. So this is measuring the transition between particular states. And we measure the kinetics with various methods. So we measure the atmospheric transition. And for example, here a set of mutations. And you can see for quite a few of them that are non-changed, but for the mutation with the red circle, which is the same mutation, by the way, that we used in the previous slide for the electron microscopy. There is no difference between the minus calcium and the plus calcium. So this mutation affected the transition from the inhibition to the activation. We measure calcium dissociation. So of course, calcium is very important for the activation of the heart, but also calcium needs to dissociate for the heart muscle to relax. And so we measure the rate of dissociation for a number of mutations, and you can see for So for a number of them, there are no changes, but for one of them, there is a substantial reduction in that dissociation rate. So overall, what we found is that for these different mutations that we analyzed, there is no single unifying mechanism that can explain the pathophysiology, and that's the take-home message. There is no single unifying mechanism. What we believe is that there are a number of different mechanisms and a number of different molecular perturbations that start the process of pathology. And of course, at some point, for the cardiac muscle, when the heart, so initially the heart, of course, is able to cope. The heart has a certain reason why, to cope with the changes. But at some point, it's unable to cope, and then, of course, it goes in what is called cardiac remodeling, which, of course, trigger either hypertrophy or dilation. So what we think is that there are a number of pathways that precede this final pathway, which is the remodeling pathway. So I'm not sure how much time I have, but so in relation to therapy, what I want to tell you, 5 minutes? No? Not one? Okay, 1 minute. So very quickly. I'm going to just tell you that, with the genetics, you identify a lot of mutations, but over the last 20 years or 25 years, there is not as much progress in the development of drugs. Okay? And this is not just in cardiovascular diseases, even in cancer, even in other diseases. And I think because maybe we are not given enough time to understand the mechanism of these pathologies. So in relation to the heart, up to 2020, there was no drug that targets contractile proteins. And there are a lot of drugs that basically just treat the symptoms, just try to manage the symptoms. But in 2020, one first drug that targeted myosin, activated basically, the myosin was a drug that targets the contractile protein. And so we wanted to look at drugs, and up to today, there are no drugs that target the actin filament or the troponin system at the contractile. And so we wanted to do that, so we screened a number of compounds, and we identified 3 compounds. And so these 3 compounds here, and we studied them, and we identified this one on the left, the NS5806, as one potential candidate, where you can see there is a difference in the calcium sensitivity of the stimulants, while the other 2 compounds have no changes. So I'm going to go quickly just to basically show you that we identified where this compound binds on the protein. So this work is published if you want to have more look. And so right now what we are doing is, of course, so we are trying to see if this compound works actually not just on the isolated protein, but on the whole microscopic system. So we're trying to do that, but of course there are some challenges because we have yet to have a full normal structure for this complex, and that's what we are working on. And thanks very much. So the important slide, of course, is to thank the people. So many thanks for So if I can have that next slide. Anyway, so there are a lot of collaborators both from King's College and from South Korea, from Boston, and of course my new collaborators from here and from Japan. And then thanks very much.
Thank you so much for this great presentation, also very exciting information. Because we don't have a lot of time, we are gonna maybe ask a few questions. Let me start with Prof. Mazen. You gave us a lot of informative information about the ecosystem of biotech and why it's important. And well, maybe what is the situation right now in the kingdom? I believe the ecosystem is so important as it is one piece in different segments. We cannot just focus, for example, in the research center or in university. I think we'll have innovative products. There is like a VC, we have regulatory bodies, we have private governmental partnerships, and also we have the CRO, the CMOs. So from your perspective, Tomas, and your long experience in this field, what do you think the most important thing we need here in the kingdom right now? We have to focus on it. For example, do we need more of the research centers, or we need more of the CDMO or CROs, or we need more partnership with the private and governmental sectors, or VC, something like that?
Honestly, thank you very much for that interaction subject. My feeling is we need to do two things. One is to create a group that brings all these entities together, so we don't have any working environment for all these entities to come under one umbrella. And two, we cannot celebrate a quick win. And this is, I think, the problem. We see something, we celebrate it, we make it a big deal, and this becomes the end of it. This is useful maybe in other industries. In our industry, if we don't take something from discovery all the way to conversion, not even full deployment, then we should celebrate. Before that, any celebration, I feel it gives you a quick feedback and success, but it kills the motivation to bring things forward. The success is not by winning one stage. Success is to get all the stages complete and come. I think we should not we should try to push back on celebrating quick wins and forgetting the journey.
Thank you, Dr. Martin. Just one quick question. I cannot stop myself to ask. So you did a lot of good jobs. With the Saudi Vax, a lot of good initiatives, and now with the Faqih Group. So can you just give us some of the initiatives as from the private sector of this ecosystem?
Honestly, we're trying to close the gap related to healthcare data. We believe that this is one of the top care issues. The access to data has been always a complicated business. So we're setting up a system allow easy, transparent access. Hopefully, help both researchers and understand the situation to better us and also collect data for usage. That's the biggest work we're pushing. We just announced our center, Center of Excellence, and part of that, we will have a full platform with 47 years of data that we has together with an AI analytic capability, all in anything as a researcher, just join that and be able to, of course, with the governance system, national biotech. This data will be anonymized, so there's no identifiers for patients. one. And two, second, which we're pushing is to be one of the leading clinical trial units in the private sector. We're working closely with the government entities to deploy this, more agile, more speedy, more supportive for the private segment, and it will be across all our centers, both hospitals, clinics, home healthcare, telemedicine. That will be not only Saudi and UAE, so it will be one approval the entire system. I think that will be hopefully going to support the ecosystem.
Thank you so much.
Mr. Ayman?
Okay, because of the time.
So, any other questions from the audience? Right.
صباح الخير للجميع، يعطيكم العافية. أخوكم رؤوف شايع مالكي. بس كان ودي أطلع عنوان مع الدكتورة فالح، إيش هو التارجت حق التجربة تطوير الفاكسين من ناحية سيبتوتروبيزم مختلفة الفيروسات حقت كورونا في التروبيزم حقها في الانتري وفي الريسبتورز. فهل الطريقة يعني أنتم تبغوا توصلوا يعني انه هي في الفاكسينز الموجوده الان يستخدموا السبايكس كبروتين او كمسنجر ار ان اي بحيث انه ينتج بس انه بارت اوف الفيروس. انتم تحاولوا تغيروا في التروبيزم على اساس انه تاخذوا الفيروس كامل. ودي ايضاح. والبارت الثاني هل هو يعني رايح هنا الفاكسينيشن بريفنتيف وطريقه الانتي بوديز اللي انتم رايحين للباسف كوميونتي ثيرابيوتيك. هذه هي الفكره. يعطيكم العافيه، شغل ممتاز.
يعطيك العافيه. بالنسبه للبارت الاول من البرزنتيشن انا حابب بس في الفقره الاخيره من سؤالك الديكور ريسبتور بريفنتيف كانه انتي بوديز، كانه مونوكلونال انتي بوديز. الانتي بوديز يستخدم كتريتمنت او ممكن لو كان يعني تاخذ قبلها بشهر او شيء يفضل في السيركيليشن از شورت تيرم بروفلاكسس. فالديكوي ريسبتور كانه استخدام الانتي بوديز، بس الفرق بينه وبين المونوكلونال انتي بوديز انه اذا الفيروس طلع اي ميوتيشن سكيب ميوتيشن معناته الفيروس حيكون لسه انفكتد ما فيها مشكله. لكن عكس لما المونوكلونال انتي بوديز حتحط انفستمنت بروسيس ديفلوبمنت تاخذ منك سنين الفيروس within couple of months تطلع ميوتيشن سكيب ميوتيشن نو فاليو. فهذه كانت الميزه للديكوريس. لها عيوب انه لو طلعت انتي بودي دراج انتي بوديز اجينست الديكوي ريسبتور معناتها جسمك حيهاجم نفسه. فهنا الكواليتي باي ديزاين لما تسوي بروسيس ديفلوبمنت للباي مانيفاكتشرنج تحاول انك تطلع برودكت يكون as soon as possible to avoid generation ADA. بالنسبه الشق الثاني من سؤالك بالنسبه للسيبس فاكسين احنا اخذنا الار بي دي الريسبتور بايندينج دومين which is the most immunogenic part of the spike proteins اللي هو حتى لما يصمم الفاكسين يسموه بيلد على الار بي دي. اغلب الانتي بوديز neutralization antibody binding receptor binding domain. فهو اخذنا الريسبتور بايندينج دومين fused with glycine serine linker with the MC. So basically it's used as a prevention.
Well, there is an online question to Prof. Mazen. From all developments that already started, what can be envisioned that by 2030 we'll have our first biological product from end-to-end?
I think already with the end-to-end, I don't think it will be available by 2030. But development to commercial, inshallah, will be at least 2 biosimilars in the market. Development to commercial by 2028 and by
طيب معلش أنا I know there is a lot of questions بس البيج بوس يصير خلاص done decisions أعتذر لكم و I know يعني السيشن كانت أكثر من رائعة وجود الدكاترة. Thank you so much for your attending and thank you so much doctors for this great presentations and information. I really appreciate being with you here today. Inshallah, we're looking for the next session. Thank you so much.
We'll resume in 10 minutes session.
I know there are many questions you want to ask. You have them captured anywhere and has
moderator فيكم بروف مازن حسنان. غني عن التعريف ما شاء الله تبارك الله, he has given a very insightful and inspirational talk for the biotechnology initiative industries. Prof. Mazen is the Vice President for Research and Innovation at KFMRC Group, a large private healthcare provider with comprehensive services and a leader of academic medicine in the private sector. He is also the founder and managing director of Saudi Labs, a pioneer Saudi biotechnology company responsible for developing and manufacturing high-quality and affordable vaccine and biopharmaceuticals. He is a professor of surgery, hepatobiology, and solid organ transplant surgery consultant, and an adjunct professor at the Department of Pediatrics at Baylor College of Medicine. Welcome, Prof. Mazen. I would like also to introduce our second moderator, Dr. Zara Maghlabi, who holds an assistant professor of biotechnology position at King Abdulaziz University and the head of gene therapy unit at the Center for Innovation and Personalized Medicine. Her research focuses on viral vectors for gene therapy, aging, and epigenetics. She also leads the Medical Laboratory Sciences Department, Quality Assurance, and Academic Accreditation Committee. Welcome, Dr. Zara.
Thank you, Dr. Khalid. Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everyone, and welcome to the second session of today, the future of biotechnology in Saudi Arabia, held under the broader theme of the conference, improving quality of life through research and innovation. Saudi Arabia is entering a transformative era in biotechnology, guided by its national biotechnology strategy. This ambitious roadmap aims to establish the kingdom as a regional and global hub for biomanufacturing and medical innovation. By prioritizing vaccine development, scaling up biomanufacturing, and expanding a national genomic database, this strategy is unlocking opportunities and aiming to enhance the health and well-being of the population. Academic institutions are at the heart of this vision, as we have seen throughout the conference. Research is already translating into clinical impact, and new partnerships are forming across academia, industry, and pharma to build an integrated biopharma ecosystem within the kingdom. Today, we are joined by a distinguished panel of experts who are at the forefront of scientific innovation, driving real-world impact across Saudi Arabia. Please welcome Prof. Amanda Luzi, Professor of Science of Bioscience and Chair of CAS, Center of Excellence for Smart Health in Saudi Arabia. Before his tenure at CAS, he spent over 20 years at McGill University, where he held the position of Professor of Molecular and Cellular Biology in Biochemistry Department. He earned his PhD in Molecular Biology in 1998 from University of Montpellier, France, and completed his postdoctoral training at Yale University from 1998 to 2001. Professor Galluzzi is a member of several national and international funding agencies, including CIHR, FRSQ, and NIH. Also, please welcome Dr. Muhammad Al-Amari. He is a leading scientist and entrepreneur at CAS and the General Manager of Applied Genomic Technologies Institute, specializing in pharmaceutics, genomics, and translational medicine. He holds a PhD in clinical medicine research from Imperial College London and has founded several biotech startups. Also, please welcome Dr. Muhammad Al-Falih, a biotechnologist with a PhD from University of Queensland, specializing in biologics technologies. He previously served as an associate professor at KAU. Dr. Al-Falih also directed the biomanufacturing training program aimed at building local capacity in biopharmaceutical production. He later transitioned from academic to public service, joining the Ministry of Industry and Mineral Resources as a biotechnology expert. Last but not least, please welcome Dr. Maram Al-Utaibi, is a distinguished healthcare leader and molecular genetic expert, currently serving as the CEO of Health Support Services Center at the Ministry of Health and CEO advisor at Health Holding Company, renowned for establishing critical healthcare infrastructure, advancing genomic research, and driving transformative initiatives during the COVID-19 pandemic, and was honored with the WHO UAE Health Foundation Prize in 2024. Thank you.
Thank you very much for joining us here today. This is an exciting time for everyone. With a dedicated day for biotechnology. So we have amazing panelists. I will start with KFMRC, since you're the last to sit down. From your perspective, how do you see as a strength that is today existing in the Saudi ecosystem that hopefully will help us to build biotech industry?
شكرا دكتور مازن. الصوت واضح؟ واضح؟ Thank you. شكرا دكتور مازن وشكرا للزملاء، صراحة سعيدة تواجدي معكم. صراحة في something catch my eye and I have to say it. ما شاء الله, the number of females in the room is very impressive. I look forward to seeing you girls part of the leadership team to science and technology and in medicine as well. Going back to you, Prof. أعتقد إن وصلنا مرحلة جدا ناضجة, maturity level of the ecosystem, من ناحية النضج في الأدوار, الفندنج, policy makers, etc. صراحة الحراك القائم الآن في البرايفت سيكتور مدهش الصدر. يمكن سمعتوا كثير initiatives from different companies, وراح تسمعون أكثر. They are willing to steer the wheel. I love that the private sector is actually taking sort of the first initiative to ignite, I say, the ecosystem. It gets me personally excited. I know numbers of companies that actually نسمعها ونقول is it true going to happen? I don't want to name صراحة names, but there's GSK that they're announcing a big صراحة funding. يمكن راح نعلن في المؤتمر القادم للمختبرات بعد أسبوعين. I was talking to the Prof. Mazen on Faqih and their innovation center. It's exciting time to see how can we all of us help. كونها policy maker كونها وزارة أو عدة وزارات, كونها جامعات, the number of researchers, as well as the private sector, right institutions that we have in the kingdom. أعتقد now, Prof, we are أعتقد at the age of a new chapter, if I may say. شكرا.
يعطيك العافية. شكرا. Always exciting to listen to you. Dr. Mohammed, maybe your next from an ecosystem perspective, what are the most exciting initiatives that are on the ground or being kicked off or going to be kicked off? You think will be contributing to the ecosystem development?
بالنسبة لوجهة من ناحية منظومة الصناعة، خلينا نقول مو وزارة الصناعة، لأن منظومة الصناعة تشمل وزارة الصناعة، الإسعادية، صندوق التنمية الصناعي، هيئة مدن، local content, etc. التصدير، الصادرات وهذا. كل يدرك من هذه عندها حوافز، والحافز هذا طبعا تعرف. بس لما ترجع بالنسبة للفارما سيكتور، احنا اليوم نتكلم عن بايوتك. البايوتك لها خصائص أخرى، تمام؟ طيب كيف نقدر نوصل للحوافز اللي تقدر تحفز قطاع البايوتك؟ حتى قطاع البايوتك لما نطالع فيه totally different. نتكلم biologics, antibodies, biosimilars, different vaccines, insulin, CGT, plasma، كل واحدة لها خصائص. فلما نيجي نتكلم عن الحوافز، احنا طبعا center of government recognize إنه في gap بالنسبة للبايوتك biopharmaceutical. فهذا الأساس صدر قرار إنشاء لجنة توطين صناعة اللقاحات والعلاجات البايوتك برودكت. اللجنة طبعا تشتغل على بناء استراتيجيات. اللجنة هذه لجنة وزارية برئاسة وزارة الصناعة وتحتها وزارة الاستثمار ووزارة الصحة، وبمشاركة أعضاء وزارة الصحة ووزارة الاستثمار هيئة الغذاء والدواء والمالية. تطلع قرارات وبناء استراتيجيات على أساس إنه نعرف كيف نقدر نمكن القطاع هذا وتكون كلير باث للبرايفت سيكتور. واللي دعم هذا اللي هو وجود الناشونال بايوتك استراتيجي، which is act as a north star لتنمية القطاع. فنرجع مثلا في وزارة الصناعة مثلا حاليا طلعت مبادرة الحوافز المعيارية تقريبا 10 بليون ريال حوافز معيارية لكل الصناعات. الحوافز المعيارية دي هي زي ما نقول جرانت لتنمية الصناعة لجميع القطاعات، وعلى أولويتها هي صناعة الأدوية. تجي اللوكال كونتنت عندهم الحوافز اللي خاصة بالتوطين، البرايس بريفرنس وإلى خلافه. هيئة مدن عندهم مثلا المصانع الجاهزة السبسيديز بالنسبة اللي تقدر تحفز القطاع الخاص انه يقدر يستثمر.
يعطيك العافية. دكتور محمد، على يعني let's talk about CACS. It's new يعني new and renovated shape. How do you see the public labs really supporting the ecosystem? Where is the public sector laboratory capacity play in this ecosystem development?
When we talk about our national lab, which is based in King Abdul City for Science and Technology, we are making now the ecosystem for the biotechnologies, and we try to make this lab is open for specific needs and based on the requirement. But just I would like to highlight about the ecosystem, the biotechnology ecosystem, and what's the plan from the government to make it Riyadh as a regional hub for biotechnology? Well, in fact, what we are doing now is with other stakeholders, we are building the ecosystem for biotechnology. So we are doing it's like regional hub with other stakeholders like Royal Commissions for Riyadh City, King Faisal Specialty Hospital, the Ministry of the National Guard, CACS in the middle, and King Saud University. So we're trying to make these connected together and as a complement. And in the middle of that will be the biotechnology hub. This biotechnology hub will be we have a plan to attract minimum around 200 biotechnology company between 2030 to 2035.
This is very exciting. But to you, Prof. Ahmad, I think everyone sees the exciting work CAUS is doing. Where do you see CAUS is leading in the work in the private sector industry? Where do you see this gaining plan in the biotech business side?
So, in the biotech, I will thank you first for the question and for the invitation. And this is giving me an opportunity to talk about CAUS and the strategy. During my presentation, I have part of it, but to summarize, 2 years ago, almost 2 years ago, CAUS announced, the Crown Prince announced from CAUS, he's the chairman of our board, until a couple of weeks ago, changed recently. And we are lucky we have his brother, the Minister of Energy, and his chairman of our board, so we are continuing on the same trend. So the new strategy CAUS now needs to continue leading in science, but also make impact, meaning almost give back what the country gave to CAUS. So, and to do that, CAUS decided to focus on its research capacity for translating or transforming the basic science discoveries to impact. The engine we established 4 centers of excellence. I will describe the Center of Excellence for Smart Health later during my talk, but the idea is taking our scientific basic science and those discovery and make impact. The other mandate we have as Center of Excellence and also National Translational Institute, that's another engine for CAUS and as a result of this new strategy, is while taking these discovery through the TRM sector to go through R&D and patent, but also further commercialization, build partnership. So we are, one of our missions, in fact, the existence of our center, the one I'm leading and the 3 others, is really how much or how many real partnership we built, we established. So our objective is to be partners with the industry. We started together, if you remember, 4 years ago with Saudi banks and in fact that effort, I'm sure you heard, now we've been given the fund that we were hoping for a few months ago to establish the first GMP facility for the country that was credited Mazen and his team started in Gauss in 2019 and left us with really the foundation for that. So we are continuing that. Then immediately Gauss will be playing a role, inshallah, from the biotech is to establish training programs. We discussed that also with Dr. Mazen, but now there is a big, around this GMP facility that will hopefully see the light within the next 2-3 years. We are establishing immediately a training program because if we do not prepare the biotechnologists or the expert in biomanufacturing, those all projects will not see the light or will not be sustainable. I think Dr. Amaram, remember when we visited 2 years ago in Boston with RBC committee, every company, big or small, we meet, who will be working in Riyadh Biotech City or NEOM. So we need to build a human capacity. So those two activities need to go hand in hand. And that's Scouts hopefully playing a role, able to play a role there.
Thank you, Dr. Ahmad, for highlighting the importance of collaboration in biotechnology field. Going back to Dr. Maram Wazeli, in your role as CEO of the Health Support Services Center, how do you bridge the gap between cutting-edge genomic research and everyday clinical practices to ensure the innovation directly improve patient care?
Tough question.
Sure.
Actually, it's very tough. We are in the kitchen, if I may say, of how can we make it happen. As we are speaking, we're trying to figure out a way. Is it a task force? Is it a committee? Is it sort of a tool or a vehicle to enable us to answer your question? Do I have the answer now? I would be lying to tell you. Yes, we do have it, and we will do 1, 2, 3. We're trying to understand how can we best serve it in terms of innovation and others. I was in a heated topic yesterday on this, so we're trying to figure out on how can we enable young startup innovators, give them access to genomics and genomic data. Where should we host that data? How can we help researchers as well have access and innovate and get further. يمكن الدكتور عماد ذكر شيء بسيط من زياراتنا is eye opening. Most of the innovators are young people like you. And لما جاية من ستانفورد, they're young. They just had the right tools. Yes. سمع عليكم you will have the innovative ideas or healthcare sector. The genomic sector is full of problems. I'm sure there's a room for improvement, huge room to move forward. يمكن من أهم الannouncement اللي صراحة makes me excited for genomics and moving forward is the اللي قاعد يشوفه الآن. I'm sure you guys are all aware the collaboration between big tech companies and genomics. NVIDIA with Illumina and others. There's no brainers that tells you where the direction is heading. So maybe I will answer it better next conference with examples.
Inshallah, the same panel after 5 years, you will have good examples.
Inshallah.
Right, Dr. Muhammad Al-Fayr, how is MIMR supporting the growth of local pharmaceutical and biopharma manufacturing capabilities?
جواب اللي يمكن قدمته الجروب مازن او الاستاذ نيفي مور. الجلوبال ماركت للفارما تقريبا 1.4 تريليون. السعوديه تقريبا $11 billion, which is the biggest in MENA. Expectation in most can يوصل spending up to maybe 18 billion by 2028. No one can, on long run, it's hard to generate. فاحنا عملنا اناليسيس صوره المصانع، نشوف البيلدينج، نشوف الكابابيلتيز، البايب لاينز. بعدين عملنا سكرينينج على two ways: فوليوم فاليو. ممكن بعض الدراجز هاي فوليوم بس الفاليو قليله، لانه انت في النهايه تبغى الايكونوميك سايكل تكون شغاله. بالنسبه للفوليوم ال 7% تقريبا كيجر increase volume, which indicate this is good capabilities building or ramp up. بالنسبة للفاليو also 7% but fluctuating. لما تجي نطالع السبب fluctuation introducing new modalities للbiotech. يعني تتبع capabilities small molecules try to reduce a gap in an import والexport أو local consume بس يجي دواء واحد like Cascade 1 كم؟ 1.2 billion لا 1.9 ولا 2 million dollars الانجكشن. some people think انه في انترا injection بروح، لا واحد يعتبر injection بس عندك hospitalization maybe 8 months that's extra cost. فعندك الريت من الاكسبورت في ضغط عليها. فعشان كذا هذا السبب اللي خلى السنتر اوف جفرمنت think of localization to keep the money economic cycle flowing. فهذا الاساس تم صار قرار اللجنه، لجنه الفاكسين باي مانيفاكتشرنج كوميتي. زي ما ذكرت للبروف مازن building strategies understand the gaps, where is the gaps, where بنشوف الجابس in general. السعودية تقريبا MENA region and biomanufacturing almost negligible. ما في few examples maybe مصر، إيران، بس ما هي يعني بالكباسيتي اللي في يوروب. China is coming strong. أمريكا، إنديا، برازيل. مع إنه المملكة موجودة في geographic location excellent, close to everything, economic stability. So we think in biotech we can play a major role in the value chain. Okay, هذا one thing. The other thing بالنسبة للprivate sector ولا حتى الجفرمنت لما build strategies ولا build business case لازم نعرف إنه biotech يحتاج high agility, high adaptability. اليوم توطن انسولين مثلا، خلينا نفرض الانسولين جاك GLP-1 how you can adapt that. Insulin injection per week how you can adapt that. Introduction of AI دحين AI دحين مثلا ما ادري اذا سمعت الخبر FDA قالوا we will phase out the animal studies for monoclonal antibodies. طيب phase out يعني ما في animal او تقليل الانيمال. They will focus على السفيرويد, spheres, culture, in vitro studies, AI driven data. Charles River lost 25% of their stock. At the same time, kept secure safety phase 1 clinical trials, how to file a pre-IND, IND, who can guarantee that? في البايوسيميلرز مثلا قال لك هو مثلا مهر في UK قال لك في البايوسيميلرز approval we don't need to include human studies, will be satisfied with the in vitro animal studies. US FDA EMA they want to reduce the dependence on human عشان يحفزوا الشركات to invest in biosimilars. Biosimilars الأوريجينال فيه حتى صار. طب كيف how they can يعني phase out the animal studies ولا phase out the human studies. there is a big debate. يعني ecosystem is need high adaptability and long term patient. Thanks.
But at least there's a roadmap for in the MMI. Thank you, Dr. Abdullah. Dr. Muhammad Al-Amri, Saudi Arabia has ambitious goals for personalized medicine. How is genomic research at CACS translating into real-world healthcare?
First, I would like to highlight that is our Saudi human genome phase 1 was officially started in 2018 after Crown Prince Mohammed bin Salman opened it and started that project. And we finished the phase 1 in 2023, and we identified after the sequence around 100,000 samples, like we found 7,500 disease-causing variant. Out of these, around 3,000 unique rare disease. And we also we identified around 1,230 rare disease recorded for the Saudi population. So what we are doing now is that we are upgrading our laboratory, and we're trying to start the phase 2 Saudi human genome, and should be will start it very soon. We are now upgrading our sequencer from the Novosix 6000 to the Novosix Plus, and we'll be sequenced around 75,000 samples. What we are doing now is for this Saudi human genome, we will try to build a national platform for the rare disease in the Saudi Arabia, and also we plan to build the national platform for multi-omics and pharmacogenomics, and we try to connect all the stakeholders together to be using with other either the pharmaceutical companies or the other stakeholders.
Thank you, Dr. Mohammed. Professor Mark, you have worked across continents and research ecosystems. How can Saudi Arabia best position itself as a global hub for bioscience innovation?
So, thank you for the question, and I think it's an important topic. We can discuss it for hours. But there are a few things for any country, specifically in Saudi Arabia, where the ecosystem is being built and it's entering this space of biotechnology. The first thing is credibility. Credibility is built with continuing doing and performing high-level science, like it's done in many universities. KAUST, of course, I mean, I'm biased here, but also other universities in the kingdom. So we should not remove our focus or even slightly compromise in the quality of the science and the quality of the output, whether basic science or practical discovery. The second thing is also build an ecosystem, and we are lucky here in Saudi Arabia. Now, like Professor Mason said in his keynote, it's the best timing to really build this in Saudi Arabia. We have the mega projects, very ambitious, like in Riyadh, Neom, and other places, and we have so the infrastructure, the ecosystem is being established as we speak. And the regulator is being also, there is a big progress. We have a SFDA. If you look now at SFDA, what they are, the policy, and they are facilitating from fast approval, the technology that we develop, to also building the policy and the process for clinical trials. And that's a space where we were lagging a little bit. And hopefully with this now push, we are getting there. So for me, it's credibility through excellency and the regulation that is flexible but keep the high standard, like Dr. Mazen said. That's really the two pillars for advanced biotechnology.
Thank you very much, Professor Ahmad. Now moving to Q&A from the audience.
How much time we left for us?
15 minutes.
15?
Yeah, we can.
Okay. We start with can we, any questions from the audience? صوتي عالي يعني. أيوة كده كويس. Any questions from the audience? Think of questions. في جاي. لا جاي.
كشف جديد عبد الله وزير وزارة الصحة my question to the KAUST is, do you have any outputs as products for market? So KAUST, what's KAUST achievement until now for this?
It's a great question, and a big part of my answer will be during my talk. The first half of my talk will be some examples. KAUST, as you probably, many of you know, was built on technology-driven university in several areas. When it came to innovation in the biotechnology sector, the last few years saw that KAUST developed a lot of tools for diagnostic, for data collection, wearables. I will give a couple of examples. And several of them are advanced in the TRM that are ready to be taken and commercialized. Some of them, I will give you, for example, Dr. Sabir Hamdan. I will give. He developed a kit, in-house kit, to diagnose for rapid PCR, one-step PCR for COVID, and we use that diagnostic kit in KAUST to this day. If somebody needs to be diagnosed by COVID, we use it throughout the pandemic, and that's SFDA approved. And based on that, I will show. I'm taking part of my talk, which is good because it's a little long. So he built now a company he called BioThor, and he developed with his technology kits to diagnose dengue, MERS, etc. And he's ready to deploy them, and he's looking for partnership. That's one example. We have other examples. I'll show you more during my presentation.
The short answer is yes.
Well, I think it's a good opportunity having this diversity in the panel to ask very tough questions. So I think it's a very tough question. And I think we need to find a way. Today, I see the government, private sector, looking at the commercial side, localization, national security. KAUST maybe, KAUST also, maybe we saw an example. But these are, to me, it's very difficult to penetrate. For anyone today from here, or from any university, anyone with a new idea, it's very difficult. People give up quickly. So if they have a new idea, they want to start their startups. And this is the reality. Forgive me if I'm very blunt in my points, but today it's very difficult to go into KAUST, if a startup coming from outside. What's the support from MIM? What's the support from MOH? What's the support from KE? What's the support from the big pharma, commercial, private healthcare? It's very limited. And people are frustrated, especially the startups, students, new professors. What can we do? What's the problem? How can we solve it? And this is for everybody.
I'll just give them time to think of an answer. We at Happy Healthcare Group, we just announced a couple of days ago, I think it's out in the media. It's a platform for startups, but this is an health AI. So if you have a startup, you have a code and you have established it, and you think it's ready for testing, validation, we're building a platform with Google support. You can actually bring your platform, you can upgrade it, you can test it. Our data is available to do the validation. We have working with RDA for governance, that includes that as an FDA, and government Commercialization Authority, and RDA. VC funding also. So we have STB, SVC, and others on top of it. So I think we're starting to build the venue to exit. Now, when technology becomes vital, it becomes a little bit harder. So I'll give you the mic. You want to start?
Yeah, I would be happy to start. I think this is a great question, and I think we discussed it for at least I've been here for 4 years and a half, and that's probably one of the main topics. And it's a valid one. Now, at least from my vantage point of KAUST, what KAUST is doing for access. First of all, with the new strategy, I think I'm hoping people start seeing KAUST is opening up to the country and coming to Riyadh, coming to Jeddah. The proof, myself and a few other colleagues from KAUST come to these conferences, etc. That's one, to engage the dialogue. Now the access, KAUST, to its credit, and I think probably it's not well advertised, there are competitions for discovery, for new idea, where there are competition with funding that are open on a yearly basis. During WISE, for example, we have last year, I think we have around 600 applicants, and the top 5, they can get substantial amount of money for to start their ideas for, and also being offered space in cows. So of course, if you have 1,000 applicants and you can accept only 3 or 4, obviously you will have 900-ish people will be upset with you. That's of course, it is normal, but at least the effort. But what I agree also with you, Dr. Anwar, is we need to work together. What Dr. Mazen said, let's remove competition. Let's see where is our strength as various entity, and put our effort together to be inclusive to the maximum. But again, we need also to keep an eye on quality. What is the killer, like Dr. Mazen mentioned, if we start advertising and publicizing every little thing we do? And this is really, that's when we lose credibility. We need to focus on quality, but also be inclusive and listen to everybody, but have a competition, but we work together. White House doing the same thing than another entity. We can complement each other, each entity do different things, but we do it together. That's
Okay, Prof. Mohammed Industry.
I will answer as a double agent.
Double agent.
I spent 6 years in university, then I moved
بس الجامعة أظن حتى قفل.
So as a minute, I will take it zoom out, then zoom in. Zoom out, the center of government recognized there is a huge gap within the ecosystem from R&D to commercialization. That's why the RDIA being formed, the NIH, to close the gap. Then the Ministry of Industry, then maybe if it was health sector, health-related product, Ministry of the Health is the biggest customer. So now there is an alignment between the different entities. Now, when it comes to funding, each entity has mandated to fund specific stream, maybe based on the TRL or based on the scope. For Ministry of Industry, we focus on commercializing products, not R&D. Having said that, to close the gap, we recognize that some of the universities, the scientists, they want to have like infrastructures, etc. We only provide in Mudun, but Mudun is far away. It's hard for the student, hard for the professors. So we established برنامج رافد. برنامج رافد. تمام. برنامج رافد تعاون بين الجامعات ووزارة الصناعة لمصانع نوعية، تعمل مصانع نوعية متقدمة بحيث إنه دكاترة الجامعة يستفيدوا منها. في ميزات كبيرة للبرنامج رافد، مثلا قروض، أتوقع من غير ضمانات، مليونز، جرانتس، تصريح صناعي، تسهيل مع الفندنج انديفيدوال. فيها ممكن منح ابتعاث، فيها a lot of things. هذا برنامج رافد. في جامعات توقع، جامعة الملك عبد العزيز وقعت، جامعة الأمير نورة وقعت. في كمان طبعا لما نتكلم وزارة الصناعة ما نتكلم بس عن الفارما، cover everything. في كمان حاضنات نمو، رحلة لكل الانتربرنور إلى ما يوصلوا بعد كده يوقفون مع الفندنج انتيتي والفاينانسنج انتيتيز زي البنوك السايلك وكذا. السؤال، بما إني كنت أنا في الجامعة إيش بتسوي؟ الجامعة برضو هي هذه حقيقة، يعني لازم takes two to tango. فالوزارات هذا بيقدموا في نفس الوقت الجامعات، يعني لو يكون في بنش ماركينج، بما إن كنت أنا كجامعة أتكلم كجامعة، بنش ماركينج الجامعات كاوس، الجامعات برا إيش بيسووا؟ to stimulate ignite الانتربرنور إنه ينجح. What kind of job أنا أعرف مثلاً الواحد اللي بيسوي ستارت اب ما عنده الكفاءة المالية، كيف يقدروا يساعدوه في التوظيف؟ كيف يقدروا هاي ستودنت بوست دوك؟ فأتوقع إنه إن شاء الله نقول إنه مع الأمور تمشي وفي سكسس ستوري، وي نيد جاست سكسس ستوري. دكتور علي يعني مثال إن شاء الله لما يكون في سكسس ستوري والناس يشوفوا النجاح I think they will follow زي ما ذكر دكتور مازن بالنسبة للأشياء related to IT. When they see success نجاح I think إنه حيكون.
طيب عشان أقفل with the positive notes I will twist صالح Professor. What is your advice to young people sitting in? What do you advise them to successfully work with CAPS and with others from your perspective?
Just I would like to highlight with the CACS. CACS, we have OSIS Park. So OSIS Park is just support with the innovation and the startup. So we have garage, but you know what's the problems? The problems you're applying for different programs by mistake. So if you have the idea, you can apply for a specific program for the startup. You have like the prototype, you have to apply for the incubator. So it's depending the program you can apply. Now we have program between CACS and Garage that include King Salman University, Faisal University, King Faisal Specialty Hospital, and Saddam University that they can apply for any idea to they started for the startup. So if you wanted to apply for any like program in the CACS, you can apply directly to the CACS. So then you can start it. So it's depending type of your idea. For example, if you have deep tech or you have some things about the technology, they have it's like one way. So the others project you can apply for different programs. So you have to decide which program. So I think this is the area you will lose if you don't apply to the specific.
And I have to say, Garage has been very vocal and positive. young entrepreneurs. However, we needed to let the spirit of the youngsters just sit back. What do you advise people?
شكرا دكتور. I'd like to be part of the conclusion. Not that the question was easy at all. I would يعني سألني دكتور مازن أعطيت مكاني نفسه. All of us at some point in time we were on their seats. I guess just be resilient. If you ask me, and if you look at my accomplishment, you don't take no other answers. Your age, I would recommend go meet, seek, build skills, skills scientifically, know what's going on, know the lay of the land. If you're ever visited CAPS, visit CAPS. There's also another position for me is شركة الصحة القابلة. So come explore. You're at a very exciting era. I'll be very honest. كنت بعماركم وكنت قاعدة مكانكم. We did not have leadership that is close to you guys, that is opening and welcoming. وبالعكس تعالوا تعلموا وشوفوا what can you do, what can you build. أنا يمكن بس أعقب على الدكتور I'll give you more positive. We can talk about regulation or policy day and night. Let's pause. Let them take their time. كونوا باللجان اللي يذكروها الزملاء or whatever. See حركات. See private sector interested at this time, at this era. Innovative. I don't know how many of you been into healthcare. You visited the hospital. There's a lot. If you are in the genomics and the genomic research, it's your era. أنا جيت من ستانفورد يقولون give us 10 years we'll dominate healthcare. We will make it a code if you want to prescribe medicine. We will put this for that. So the sky is the limit. لا تستصعبون الأمور. meaning okay ما حيرد عليا. I answer on LinkedIn. People who reach out I answer on LinkedIn. فقول يعني احنا بالعكس يعني يفرحنا. انا صراحه يفرحني لما I see young people excited. They want to learn, they want to know how to navigate. فأi'll leave you on that positive note. شكرا دكتور مازن, شكرا للزملاء. I am personally happy to see you all today.
So resilience, don't feel shy, knock the doors, don't get knocked off. في حاجة كمان؟ هذا واسطة.
No doesn't mean no. Doesn't mean come back.
It's a comment. I just wanted to be a devil's advocate. To make sure everybody knows that there is something on.
There is something going on. Like that. We'd like to thank the committee. صراحة, this is an amazing catch them during the breaks. There are some amazing leaders. They have multiple hats. They've had multiple hats. They will continue to have multiple hats. So make sure that you mingle with them and take their contacts. You never know when you're going to need them again. Thank you very much.
We'll resume at 1 to hear from our keynote speaker, Prof. Imad, inshallah, about the exciting opportunities and success stories in CAPS. See you all.
to this afternoon session, the final session of today's theme of innovation and biotechnology. I would like to start by introducing our moderators. The first moderator who we had a brilliant talk about, very insightful about the recent application in biotechnology, Dr. Hussam Tayyib, who works as the director of the Center of Innovation and Personalized Medicine at King Abdulaziz University, who also serves as an associate professor of nanomedicine and biotechnology at the Faculty of Applied Medical Sciences. Dr. Tayyib accomplished inventing and holding 3 patents from US Patent and Trademark Office. Welcome, Dr. Hussam. I would like to move also to introduce our second moderator, Dr. Ahmed Al-Basri, who holds an assistant professor in the Department of Medical Laboratory Sciences at King Abdulaziz University. He earned his PhD in medical genetics from the University of Bristol in the United Kingdom. His research focuses on stem cells and tissue engineering with a broader interest of advancing regenerative medicine and genetic therapies. Welcome, Dr.
Starting with the third session for today's theme, biotechnology and innovation. We're glad to have Professor Ahmad Kalouzi as a keynote speaker in this session. Professor Ahmad is a professor of bioscience and the chair of the KAUST Center of Excellence for Smart Health in Saudi Arabia. Before his tenure at KAUST, he spent over 20 years at McGill University, where he held the position of Professor of Molecular and Cellular Biology in the Biochemistry Department at the Goodman Cancer Center. He earned his PhD in Molecular Biology in 1998 from University of Montpellier in France, and subsequently he completed his postdoctoral degree, postdoctoral training at Yale University, US from 1998 until 2001. So let's welcome Professor Ahmad.
Thank you so much. So, thank you for the invitation, Dr. Anwar, the organizer. Really, it's a pleasure and an honor for me to be here and to share with you 2 parts of my talk. I would like to introduce some of you who don't know KAUST and also the strategy, tell you a little bit about our Center for Smart Health, and also the big part of my talk is what I do in the lab. But the main message that I would like really to leave you with is for biotechnology innovation, for innovation, translational research, everything starts with basic science. And I hope throughout my talk, I will try to really reiterate and give example of what I mean by that. So for those who don't know KAUST is 100 kilometers north of Jeddah, more or less, and was built in 2009, and it is a graduate university. So far we have around 1,700 graduate students between master, PhD, and trainees, around 190 faculty, and scientists, postdoc, and research scientists. Of course, superstar. In fact, KAUST is a mini city, it's a village, and 8,500,000 inhabitants with a family, etc. So, 2 years, I mentioned during the panel, in August 2023, the Crown Prince, who was chairman of our board, announced a new strategy of KAUST. And the new strategy of KAUST was the main message is KAUST reaches, at that time, 13 years since its inception. And now it is time that while continuing delivering world-class research, world-class basic science and discovery, now it's time to probably give back to the country and to translate more and make more impact. And of course, like many changes, we worked hard, we struggled with the change, I mean, we suffered a little bit like many institutions go through that. But I think we did it. we are on the right path. So one of the results of this strategy, of the arm of Kaust's strategy, is we established 4 centers of excellence. Smart Health, that's the center I have the privilege of leading, generative AI, renewable energy, and sustainable food security. And these centers, more or less, are the faculty are member of the divisions, but also member of the centers. Center of Smart Health, which I would like to introduce you to, have really our approach and mission is that we combine both health research, health-based activity, and technology. KAUST is very strong on technology, on computer science, AI, machine learning, and bioengineering, nanotechnology. And also, for the last 6-7 years, KAUST built a strong group of faculty in the health science. So our center is really combine both and drive programs that are based on both. That's our thing. So our center attracted a lot of interest in KAUST. In fact, I told you we are 190 professors at KAUST, faculty, 43 of them are affiliated with our center, which is a really, and so multidisciplinary. We cut across all the division. The majority of the expertise of KAUST are joined the center to really solve health challenges for the country. So here's our role. Our role is, as I told you, KAUST was built on really driving world-class basic science. We are continuing that. But the centers, one of the main missions of the center is move from the first of the idea and proof of concept and proving ideas and deliver publication, etc., move that throughout the continuum for translational research, which we take them from the validation, then the R&D, then encourage our scientists and support them to transform their patent to startups and application. And at the end, we have another arm at KAUST, was National Translational Institute or Transformation Institute. This is the arm where now is connecting KAUST with industry in a formal way, more active way. So we have also the possibility to move our products further, hopefully up to commercialization. So now our center has 4 pillars. 2 are health-related, health-focused. 1 health arm. This is the concept. Probably I will be happy to elaborate in another setup. But this is a concept where we look at individual health, not only of what's happening in our organism and our pathologies, but as an individual, as an entity as a whole. Not only your organism, but also where you live, the atmosphere, pollution, etc., lifestyle, etc. And that's a concept now really is built, established, actually this concept more than 20 years ago, and now it is really an important focus of medicine, and that's how we move on. We no longer treat, hopefully we will not treat only diseases, but we will promote health. The second part, the second arm is promoting healthy aging and the gender of medicine. This is to keep up with one of the country's priority, which is aging, and also use all the cutting-edge technology from stem cells to adenovirus, etc., etc. All of that we are establishing at KAUST. So we have 2 also other arms which focus on technology: the smart diagnostic and sensing technology and the bioinformatics. That's mixing AI, combining AI and wearable technology. We have, as I might answer earlier to one of the questions, there are colleagues for the last 10, 12 years established and created a lot of devices for various applications. I will give you some examples. And now we are combining AI for data collection, data mining, and we work closely with the Gen AI Center of Excellence. I think I was told that my colleague Paying already came and presented during this talk, but Dr. Paying, more than 2 years ago, before the inception of the center, our center started last summer, but we had Smart Health Initiative before, which I was the associate director of that initiative, on which our Smart Health established. This is MISA approached us more than 2 years ago, asking us whether can we establish a biosurveillance system in the country. So Paying is one of the world experts in, for example, in wastewater and studying pathogens in water. She already developed a project that we supported. For example, here, teaming up with Mecca University and collect water from the plants there. And I already identified, and this is she published this, unique antibiotic-resistant genes handled by pathogens. The other thing, we teamed up with the Jeddah International Hospital, where we collected sewage from airplanes. This is 18 months ago we started this, and Paying and her team did a lot of sequencing, and now I can say that we can identify incoming pathogens. The application of this is a huge for biosurveillance, for Hajj, and for incoming tourists, etc. The second one is you heard me talking about it, is Dr. Samir Hamdan. Now, this is a typical example of an expert. He's really basic science. Samir Hamdan is a world-class scientist working on replication. Many of you know replication, and really what his expertise reconstitute the big machinery of replication in vitro. Thus, he built his postdoc at Harvard. He was one of the founding members of KAUST. When the COVID-19 hit, there was a call for many scientists at KAUST to help address COVID-19. Samir answered that call and built PCR kit, because for him it was easy to produce the two enzyme needs for PCR, and he optimized the way to make it in one step. Less than 1 hour, you can diagnose whether you have COVID or not. And then, as I said, we used that kit to this day. However, Samir went further. He established a company last year that he incorporated, and he also developed kits, PCR-based, mRNA-based kits to diagnose all of these hepatitis B, C, HIV, MERS, etc. And now he's talking with several institutions in the country for validating these kits and then for commercializing both in the country and outside. Now, Dr. Dana Suleiman, she's our rising star. She's an assistant professor, Saudi, trained at MIT. She joined KAUST more than 3 years ago. And Dr. Dana recently, among other things she built the system by next generation of mRNA-based biosensors. She's an expert in biomaterial and also in making biomaterial compatible with organic material to, in this case, create an app on our phone or a device on our phone where she built that biomaterial-based chip linked to DNA, and that can really sense or identify very little amount of microRNA that could be biomarker, for example, for cancer. Liquid biopsy can be put there. Very quickly, you can identify the set of microRNA, and you can say yes or not, probably you have cancer. And this is that got a lot of attraction. She is now discussing with a couple companies in Europe to license this and move it forward. Sahika and Stefan, some of the colleagues here know them both. They developed during COVID era this technology where you can identify very, very bioelectric protein sensitive detection. I mean, it's a device where you can detect few molecules of biomarkers. They applied it for COVID, and now they're applying it, for example, for neurodegenerative disease such as Alzheimer and other indicators. And these are based on nanobots. Nanobots are small, big, similar to antibody, but are very small, very easy to produce, and they use that technology to use it for diagnostic. Sheng Gao is a colleague of mine, is an AI expert with application in health. He published a few months ago what he calls Skin GPT. Skin GPT is an algorithm that you can connect on your device, iPhone, take a picture of your face, and it can tell you very quickly what type of skin lesion you have. And again, he attracted interest by several companies and is in discussion to commercialize this. Now I'm moving to tell you what's the connection me to smart health and to cows. So I moved to cows 4.5 years ago from McGill University, and my training since my PhD work, I was interested in mRNA biology in general, but the role of players in the mRNA field. As you know, I don't need to introduce you to mRNA. In fact, even in my family and my friends who are not biologists, you say mRNA like you say computer science. So it's like you looked at, oh, wow, like genius. So, however, the vaccine that have been developed, or a majority of us have in our arms, could not be developed except if we did not understand this process, how proteins are made in our cells. That took over 60 years since the discovery of mRNA and double-strand DNA to reach that level of understanding. And just to summarize quickly, as you know, genes are in our cell nucleus. They produce mRNA molecules via a process called transcription, and that molecule moves from the nucleus to the cytoplasm, and in the cytoplasm where there is the protein production machinery, the ribosome, etc., and we have proteins produced. Now, like you heard earlier, the mRNA technology indeed existed in early 2000. In fact, Moderna was established around 2005, 2006 by colleagues, one of them from McGill, so I happen to know. And believe me, Moderna was a very small company, very small, and they are always there in our meetings, etc., have booths until 2018, 19, where things changed. So that would not have been, Moderna was applying FDA since 2010, 2011, like you heard this morning. And it was always rejected for one reason, because the mRNA molecule has two problems. It's unstable, that can be produced, the mRNA molecule produced in vitro, and also trigger immune response. Because of these two people, and they got the Nobel Prize 2 years ago, as you know, that was solved. And the moment that was solved, gave the possibility that we can now introduce mRNA molecules that we can produce exogenously to our cells, and we produce any protein we want. And that, in the case of COVID-19, the spike protein, and we have immunity. And now, of course, I mean, there are many applications that you know. Now, my lab focus on these players, RNA binding protein, because of course an mRNA molecule is very fragile, very flexible, but very fragile, and cannot be moved from the nucleus to the cytoplasm and be processed. There are a lot of steps to process the mRNA to make it readable by the protein machinery. These RNA binding proteins, these are various types of proteins, more than 3,000 that have been discovered since the 80s, and their role for a long, long time stayed as a mystery. So my lab, when I built my lab in 2001, 2002 at McGill University, I took with me that project, and the question was, what is the role of these proteins in mRNA biology in general, but in particular, can it have an impact on our health? So I use, as an example, muscle regeneration and muscle adaptation. In disease, I focus, say, in cancer, like everybody else at that time, but also muscle-related pathology called cachexia. I will tell you about it a little bit. And of course, I had an interest of mine for a long time, the role of RNA binding protein in aging. So as an example for health, the role of these RNA, and I will try to show you at the end how all these research, which now more than 25 years of research, actually, it's hard for me to say it because I still in my mind 20 years old, but in my body, I mean, so I realize how old I am when I say this. So I took muscle for that in 2000, 2001. Muscle formation and muscle function was really well dissected at the molecular level, except when it comes to mRNA. Anything after transcription and before translation were almost black box. So I said, that's a niche I can develop with my expertise. So I built literally my career on this protein. I started working on this protein during my postdoc. It was just discovered a few months before I joined the lab at Yale, and asked the question, what its role? HUR. In fact, I was so close to this protein, like when I met my wife in 2000, who became my wife in 2007, then we decided to get married. When we had our firstborn, when she was pregnant, I tried to convince her to name my boy HUR. Thank God she said no. Huh? So, well, just to tell you, so this protein was, of course, its role was literally unknown, so I was lucky I developed all our understanding in cell in general, cell adaptation, but in particular in muscle cells. So muscle cells, muscle regeneration or muscle development happen the following steps. Imagine when we have injuries, there's a group of satellite cells that are hidden here, stay dormant normally. If you have an injury, they are activated, and the purpose is to heal the muscle. Then they develop, become myoblasts, they grow for a few days. And when you reach critical mass, they start fusing. And that fusion, you have nice fibers. In my work of the last, probably, let's say 20 years, literally, this is some examples. My lab showed that HUR can be involved in the first step of myogenesis, or muscle fiber formation, as well as the second step, with different functions. Keep this in mind. It can promote, stabilize target messages, but also can degrade some of them, and can participate in the translation. So it was really, in fact, it's like P53. Some of you are familiar with P53. P53 had multiple roles. HUR is P53 of post-translation regulation. So the question we asked, after we demonstrated for 15 years its role in the cell, we used several cell lines, etc. And then we came a time where I needed to switch to become a physiologist, move to use animal models. So we asked whether the function of HUR, is it true that regulates muscle in vivo? So we took mice, like everybody does, and created the muscle-specific knockout mice. And the reason, because total knockout of HUR, the mice were not even born, they die at 10.5 during embryogenesis. So, of course, that work, the knockout and all the results of the publication took literally 10 years. So, so one of the big results of that work is when we knocked down HUR from muscle, I thought the mice will die, will not burn based on our in vivo data. No, mice are healthy. In fact, the mice, you see that that mice is more resistant, has a better endurance. And so, in fact, when we published this work, the conclusion of the work was that when you remove HUR, you have oxidative fibers, and that's what marathon runners have. That's one surprising result. The second result, so then, since my lab, I told you I'm interested in the role of RNA binding protein. We know HUR, we established, other labs worked on it for a few years, confirmed everything we observed. So we continued trying to identify other RNA binding protein. We found that, and this is work by Brenda Sanchez. Brenda actually did her PhD, joined my lab in 2012 at McGill, did her PhD with me, and she did not have enough suffering working with me. She followed me at KAUST. Now she's continuing working with me at KAUST. So Brenda also discovered that HUR can associate with another protein called YB1. And we demonstrated, after a couple of years of work, we demonstrated, and this work, big part of it was done at KAUST, so that YB1 can form a complex with HUR, and they regulate, stabilize target message like myogenin, MyoD, and other key pro-muscle formation players, their mRNA to produce the proteins in specific sites, that site, on their 3' UTR. And then we published that a couple of years ago, but that's triggered a question, because in the mRNA field, we know how mRNA are degraded. The machineries have been dissected for the last 20 years. We know a lot about the decay machinery. We know how mRNA is translated and become protein. However, when it comes to mRNA stability, the only thing we know this day is that protein bind specific sequence and you have longer half-life or protection. We don't know anything else. So what we decided, Mariana joined my lab a year ago, and the question now they are working on it together with Brenda to identify the mRNA stability machinery, or what we call stabilism. I have two other students. I have several students. I'm lucky. I mean, when they joined CAS, I built my lab fast relatively to the time frame needed for building a lab. So I have very talented students. Victoria and Rina joined my lab a year and a half ago. And each one, we discovered this protein. It's long. I mean, for sake of time, I'm not saying how we came across that this protein can be relevant to muscle formation. So DDX3, this is a helicase protein, and Renan is working on Maton3. And they, of course, I'm showing you, I'm talking about them because we demonstrated after, I'm showing you only a couple of data, but they have a full study that we are preparing them for publication, for submission, actually, in the next few weeks, hopefully. So one of the things Victoria did, for example, you knock down DDX3, you see the control, and you use siRNA to knock down DDX3, and immediately you see you prevent muscle formation. These are markers in immunofluorescence show you see the MOC and the aside control. You have nice differentiation. These are C2C12 cells. They grow in the lab, and when they reach confluency, we starve them, and 2 days later, 2-3 days later, they start forming these nice fiber, as you see here. So if we remove DDX3, we block that process, and then mechanistically, how DDX3 promote muscle fiber formation? It is by acting on this signal transduction pathway called Wnt signaling. It's a very important signal transduction pathway for muscle, but also for other regulate cell progression, other cell types. So when we knock down DDX3, we affect at least 5 members of the Wnt signaling, and that's the reason why we prevent muscle formation. To Renat, it's basically the same observation, except Maton3 has a different role. We knock down Maton3, we prevent muscle formation, and Renat found out that now Maton3 role is to promote, stabilize, and promote the translation of players in the mitochondria. I'm sure majority of you are familiar with mitochondria. Mitochondria is the organ that produces energy. It's a key player in our cell metabolism. And when we knock down Maton3, and she has beautiful data recently, electron microscopy, etc., showing that effect. And so that's the conclusion. So we have now 2 additional RNA binding protein play a role in promoting muscle formation. So now when it comes to disease, obviously my interest to the role of RNA binding protein in disease cannot be far from muscle. So we asked the question whether HUR, for example, that's, I go back always to the origin of my lab. So whether HUR can play a role in a syndrome called muscle wasting or muscle atrophy. So one of muscle atrophy can have different outcomes. Sarcopenia, or elderly have a chance to start losing their muscle mass. That's sarcopenia. It's a slow progression and slow loss of muscle mass. However, in response to disease such as cancer or viral infections, you can start losing your muscle very fast, within a few months. In fact, when many of us unfortunately know cancer patients that start losing their muscle mass that become cachectic, and they are sent to palliative care waiting for the end. There is no cure for muscle wasting. And when you develop muscle wasting, you start being refractory to any treatment, etc. So it's a big health problem that is, to this day, it is still unsolved. So obviously, to solve the problem, more we know about the molecular mechanism, better chances we have to develop cure. So my lab worked for this, on this disease for the last 15 years, and we identified that HUR, remember, HUR promote muscle formation, but also can, also in response to disease conditions, become a promoter of muscle loss. And the way it does it, by promoting the expression of 2 players, INOS and STAT3, that has been tied to muscle wasting for at least 2 decades. Now, remember, this is the paper where we showed that when we remove HUR, we have higher endurance. And those mice, interestingly, also when we inject tumors we did a xenograph experiment and we inject them with various tumors, adenocarcinoma, for example. They are resistant to muscle wasting. So how come HUR promote muscle loss, then also promote muscle wasting? So the question now, can we target HUR as a drug for muscle wasting? There is an issue there. Before we go there, we need to understand how HUR go from being promoter of muscle formation to change and switch, become bad players and promote muscle loss. How that happened? I had this observation for the last 8 years. I could not convince anybody in the lab at that time at McGill to tackle this question. I have fondly, Muhammad, he's an MD PhD, came from Faisal University. He joined my lab 2 years ago, and he was crazy enough to say, yeah, I'm taking up this challenge. So when Muhammad started working on this, so obviously what we tested, how a protein changes. There are several ways. One way is you change the ligands, because protein can associate with other protein to do a certain function. So if you change the ligands, in response to disease stimuli, that may be explained. That's one way, but the more classic way is post-translation modification. For example, proteins can be modified with tiny signals. I'm simplifying for those, and forgive me for those who are familiar with it, like phosphorylation. This tiny modification of the amino acid that change the whole, let's say, structure of the protein or folding of the protein, and then can switch from one function to another. So we tested phosphorylation, the answer was no. And I don't have time to tell you, we tested 13 phosphomutants of HUR, and the answer was no. Also, HUR in 2002, long time ago, was also shown that it's modified by methylation. We tested that, the answer was no. However, in 2017, there was a paper showing that HUR is also modified by another modification, which is less until that time, but now it's more common, what we call PADP ribosylation. So PADP ribosylation, and in this paper, they used it in macrophages and regulates HUR through inflammatory function. So PADP ribosylation is a modification, or post-translation modification that has been discovered a long time ago, tied to DNA break. So when DNA breaks, histones are PADP ribosylated, or that is messed up. The PADP ribosylation is messed up. What's this? PADP ribosylation is you use PADP ribose with the reaction, an enzymatic reaction that is catalyzed by enzymes called PARPs. It's a big family of enzymes, and you transfer that PADP ribose on a target residue, and you can form polymers and form a mesh. So it has the end, that's reversible. That reaction is reversible. This PADP ribosylation, if you read the literature, there are 17 PARPs. Only the first 6 have catalytic activity, and the most famous ones, PARP 1, PARP 2, and pancrease. I'm saying this because there is a reason. So recently, last year, Khulud, with the help of 2 people from the lab here at KAUST, Khulud, she's a Saudi student, did her PhD with me at McGill, and she finished. By the time I moved to KAUST, she graduated, and the work was finished by Sujita and Ali, who are members of my lab at KAUST, and we demonstrated that part 1, as you see on the right, part 1 polylate HUR and promote its movement from the linkers to the cytoplasm. And that movement is very important for HUR function. And we demonstrate this in HeLa cells. Also, last year, Suad published with people from, with Brenda, etc. She published that pancreas, remember I told you pancreas, also polylates HUR in muscle cells, and is required, that modification, for the pro-myogenic function of HUR. Now, what Muhammad observed, recently, a few months ago, is when he looked to the expression of pancrease and PARP1 in vitro models. We take the tubes and we treat them with cytokine, TNF-alpha and interferon. That's how we trigger rapid collapse of these fibers within 24 to 48 hours. When we treat these muscle fibers he observed that telomerase 1, which I told you, is required for HUR to promote muscle formation. Its expression is downregulated. This is a Western blot that we assess the level of this protein, in this case telomerase and PARP1. However, PARP1 is upregulated. Normally, PARP1, the moment you induce differentiation, it collapses. It's no longer expressed. Here it's the opposite. So, what he's continuing working on, we know that telomerase 1 is needed for HUR to promote muscle formation. We don't know yet, but he's testing it with a PARP1 because now it's upregulated under cachectic condition. Is it the one that modifies HUR function so switches to become a bad player and promote muscle racing? And that's what Mohammed is doing. And the idea here, if we understand that switch, we may be able to develop molecules that will promote the good behavior of HUR and prevent its bad behavior when cachexia hits. Now, the last part of my, how much time I have? 5 minutes, it will be less. Now I want to tease your imagination. And this is where I'm very proud, and I think my best decision, the best decision of my career was to move to Saudi Arabia and to CAST, in particular, but Saudi Arabia in general. But look, add to that, think about it, if Anwar knew me since I arrived to CAST 4 years and a half ago, and I kept this enthusiasm the same. But a year ago, you all know this lady. Who doesn't know her? Do you know who she is? Rayana Barnawi, the Saudi astronaut who went to Saudi the space station in May 2023. So last year, during an event that we organize every November, I invited Rayana to be our guest of honor and to celebrate her, and CAST was very enthusiastic. It was a big celebration. And when she came, I realized Rayana had a master in molecular biology from King Faisal. Actually, she did her work in a friend of mine who became a friend of mine, Wanda. And we discussed, I told her, you should do a PhD. A few months later, she contacted me and told me, Dr. Ahmed, I spoke with the CEO of SSA, and I'm doing PhD with you. That's luck number one. So that immediately, for me, and CAST was really very happy to this day. I mean, we're so proud and happy to have Rayana as our student. So immediately now we start designing experiments. She started officially her PhD in September. We start designing experiments to address now, does HUR or post-translation modification, post-transcription regulator like HUR, etc., play a role in muscle in space. So, for example, let me give you the origin of this crazy idea. You know, astronauts, when they go to space, they lose muscle very fast, okay? And almost without exercise, within a few days, they start losing their muscle. This is just a biotech from Chris Hartman, who spent more than a year in the space station a few years ago. And you see, on the left, it's a section from his muscle. On the left, before he went to sensation, a few days later, that's how they look like. So really, the atrophy, they become smaller. However, when these astronauts come to Earth, a few weeks later, it's recovered. That said something about those satellite cells. Maybe satellite cells, something happened to them. So with gravity, lack of gravity in space, they are reprogrammed and they can recover muscle quickly. My crazy imagination, and I share it with you with all transparency, is if one day with now space become accessible and tourism, we may have hospitals to stay. Think about, we can send sarcopenic patients up in space for a couple of weeks, they come back and they recover their muscle. That's not, I thought it's far-fetched, but some people from NASA, when they heard it, they said that's not. So we take the advantage with Rayana, and that's probably, hopefully I'm pushing now. First, we can send the payload with satellite cells from patients versus healthy people, and we can see the impact of gravity, and we test the hypothesis, and we will assess the molecular mechanism. But my dream is Rayana goes back to space herself and do the experiment herself in the space station like she did a year and a half ago. And if that happens, Rayana will become the first scientist who does experiment in the space station then use those data to defend a PhD. That's what can be the first. So, and of course, there are other ideas in the lab. We are very much interested in aging, as I told you, and aging is connected to senescence. And we would love to assess whether gravity impact or trigger senescence specifically for brain, because there are a lot of studies indicate that astronauts, especially when they come back, they have problems with their cognition and or sign of accelerated aging. So that's going to be tested. And this is something really I'm very excited about. I talked about it since this happened the last few months, and of course, I'm blessed to have this team. I told you I'm lucky. I put together a team with less than 6 months, and we are the lab is up and running. And this is my team here at KAUST. And thank you very much for listening.
Thank you very much, Prof. Belusi, for the insightful and inspiring talk. Well, I'm so honored to be here in this conference, and I would like to introduce the next speaker, Dr. Hamza Banja is Associate Professor in Clinical Laboratory Science at Baif University, specializing in medical biotechnology. Her academic interests focus on the application of biotechnology in healthcare and well-being, with particular emphasis in bridging scientific innovation and community awareness. Please join me in welcoming Dr. Hamza.
Assalamu alaikum wa rahmatullahi wa barakatuh. I'm Dr. Hamza Banja. I'm Associate Professor in Biotechnology. And I'm glad to be here today to talk to you about how biotechnology are becoming a part of our each-day life, advancing health, wellness, and revolutionizing our future. First, I would like to thank Dr. Hussam Tayyib and the CIPM for inviting me to give this talk at this conference. So let's begin. So starting, first of all, I would like to ask, what is biotechnology exactly? Or what? I'm sure that during the day that you have heard many different definitions and great definitions of biotechnology. And to sum that up, biotechnology is the use of living system and living and organs to solve real-world problems, whether in advancing health or producing food or even enhancing our lifestyle. So let's go on and see how biotechnology is redefining the way we live now and in the future. Starting with health. Different biotechnology are helping us to predict, prevent, and personalize the medicine. I think many of us are trying to predict his disease, Googling symptoms every day just to make sure that he's right. So maybe in the near future, we can have this prediction using different biotechnology. The one that we're talking about right now is the personalized medicine driven by the, or powered by the AI, where large health data set like genomic, clinical, and genomic profile and clinical data of patients are being processed by the machine learning and AI for pattern recognition, which will help in tailoring the treatment and also the diagnosis. An example for this, in the real-world example for this, is where the Siemens and the IBM Watson health platforms are generating a digital twin or a biodigital twin of organs and of human bodies in order to simulate how the body is responding to the treatment even before it's given to him. Another application of biotechnology in health is the microbiome engineering. That's mean modifying the microbial community or introducing engineered probiotic to influence health and immunity. A real world example for this is the Synologic product. It's an E. coli engineered to treat the phenylketonuria disease, where the patient cannot digest the phenylalanine. So using this probiotic it's generating, it's secreting enzyme that can help in this digestion. The third application in health that I think everyone talked about earlier is the gene editing and gene therapy, where tools like CRISPR and prime editing are correcting our DNA precisely. You can see here different gene therapy that have been approved by the FDA, like the external treating the blindness and different treatment for the blood cancers. An interesting gene therapy lately that have been approved by the FDA is the Cas-GB, developed by Vertex and CRISPR Therapeutics, which is a treatment for sickle cell disease and beta thalassemia, where using CRISPR is helping in reactivating fetal hemoglobin to produce normal red blood cells that are functional. So, moving from health to wellness, how biotechnology are advancing the wellness the way we go. Applications, biotechnological application of genetic based or nutrition, fitness, and lifestyle, or as they call it, nutrigenomics and exercise genomics, where our genetic variation is playing a key role in how our body responds to nutrient, or how our body responds to exercises, are being processed using different technologies in order to give us tailored and diet plans and exercise. Well, an example for this is Circle DNA, nutrigenomics, where are their platform that are offering next generation sequencing for our DNA and give us insight on how we can, and give us insights and recommendation on how we eat, how we sleep, and even how our skin will respond to pollutions and many other things. And actually, this technology are brought here, are being here in Saudi Arabia, brought by the Nanoemulsions, a product called Will DNA, is doing the same, giving a tailored diet plans tailored for our regional needs. Actually, I would like to take advantage of this and saying that I'm leading a group where we work with DNA-based, and we are searching for, and we are checking the SNPs and polymorphism that are related to sleeps and an exercise in order to see how we can get advantage of this. Second application is the anti-aging and the regeneration biotech, where scientists are using different biotechnological applications like cell reprogramming, tissue reprogramming, and autophagy enhancement, and many others to help in slowing down or reverse the aging process at the cellular level. Different startups are racing for these technologies in order to reverse the aging process, and actually Saudi Arabia is being a part of this movement through the foundation, the Health Evolution Foundation, where it's investing in research and innovation for to improve the quality of aging, adding new healthy years to the life. So let's move to the last part of my talk, which is about the emerging innovation that are revolutionizing our future. Synthetic biology. Synthetic biology is a powerful engine of the biorevolutions. The first example in synthetic biology is the molecular farming, where plants are used as bioreactor or natural factory to produce molecules such as protein, and later use them for food or for enzymes and antibodies. Where a specific gene is inserted in the plant cell, and the plant is left to grow until they harvest the crops and extract these molecules. And actually, this is brought to in our real world by the Molly company, where they are using plant as an expression system for the animal proteins and giving a way for sustainable food production mechanism. Also, the 40 protein are already reaching consumer by the plant-based protein powder. Okay, this may seem to be very controversial, but the second example that we have in synthetic biology is the lab-grown meat. The lab-grown meat, or as they call it, cultivated meat, it's where cells of the muscle from cows and chicken are being extracted and cultured in the lab with nutrient-rich medium, and then in bioreactor there and or through 3D printing, they are formulated in a steak shape or a burger shape. Actually, this is also being reached the market like 7 years ago, like the Musa meat and stakeholders food. Their aim is to provide a sustainable way and an alternative way for food production for the industry. Okay, the third example on the synthetic biology is the biofabricated fashion, where designers and scientists are collaborating to generate textile fabrics, that is the leather-like fabrics, using mushrooms or shrimps or seashells products and different biomaterial with different biotechnology to produce fibers like leather and silk. A real example in this work is the Tomtex and the Old Thread, where they have developed Milo leather. It's a mushroom. It's a leather produced from mushroom, and an iconic example is the bag design from the Milo mushroom leather by the designer Stella McCartney.
5 minutes, please.
Okay, this is the last example I'll be talking about, where microbes are cleaning and powering our world for sustainable environment. So, the living power, or bacteria that generate energy, or they call it electrogenic bacteria, where these bacteria produce electrons and protons as a byproduct of their metabolism. And when connected to an external circuit, electricity is generated. An example for this is the biological battery called Biopanel, produced by a Spanish company, where it's similar to the solar cell, but it's a biological cell, where these bacteria are used and connected to wires to generate energy. Actually, this product is already being used in smart landscape and smart buildings. The last example we have here on microbes are the plastic-eating bacteria, or the natural recyclers, where some bacteria can actually produce an enzyme called PTAs to digest, to break down the plastic and digest them later. Many different organizations are working on this in Europe, like the Carbios and Hayward, and just near here is the lab of the microbial ecogenomics at KFMRC University, where they are working on exploring the microbes of the Red Sea, and actually they have isolated plastic-eating bacteria from the mangroves of the Red Sea, and they're working to help in degrading these pollution from the Red Sea as well. Finally, this is a call of curiosity in action, where with a big innovation comes big responsibility. And as we go through this innovation and this promising new technology, we should be asking during this process and considering different ethics and asking who are we benefiting and who are we leaving behind. That's all. Thank you very much.
Thank you very much, Dr. Hamza, for this exciting talk. Please welcome with me Dr. Majid Filimban. He's a biomedical technology consultant with a PhD in stem cell biology sciences from Newcastle University at UK. Dr. Majid has a master's degree in biotechnology from the University of Queensland of Technology in Australia. Dr. Majid is an assistant professor at the Department of Medical Laboratory Sciences at King Abdulaziz University. He is also a principal investigator at the Center of Innovation in Personalized Medicine. Dr. Majid has directed several projects in collaboration with national and international universities, such as Utrecht in Holland, as well as KAUST, and leads collaboration that resulted in several patents and generating the first cell line of induced pluripotent stem cell from Klinefelter cell patients. His research interest is focusing on tissue engineering, stem cell research, artificial intelligence, and medical biotechnology. Please join me and welcome Dr. Majid in this interesting talk.
Assalamu alaikum. Thank you, Dr. Hassan, for this amazing introduction. I'm glad to be here actually with these amazing scientists and researchers. So thanks for this opportunity. As you can see, this is my talk. It's an EDNA in air, a powerful invisible gem. So let's start with what is EDNA? So, or environmental DNA. So EDNA is any genetic material that can be released from an organism in the surrounding environment in any form of biological material. So why this is important? Because EDNA in air can benefit several fields. For example, it can assist the biodiversity of the environment and also can benefit healthcare system and forensics fields and law, and support the food industry. So if you focus this on these applications, we find that EDNA can have several applications on these fields. First, starting with the wildlife monitoring and environmental biology. It can assist the biodiversity in complex habitat, and also can be a tool for detection of endangered and invasive species. Here in Saudi Arabia, we have around 125 invasive species, 70% of them birds and invertebrate. It also can be a detection tool for consumed prey after predation. This can be done because EDNA can be collected several hundred of meters away from the target area. Also, for hospital and ICUs and infection control, as we know that hospital-acquired infection can affect 1 in 10 patients worldwide, and EDNA can help to investigate the cluster of these events, and also to map the pathogen transmission through this technology. Also, it can help to validate the air handling and the filtration system of the healthcare facility, as well as to verify the effectiveness of the quality control and the infection controls in this facility. In forensic field, the EDNA in air can help to detect human presence at clean scenes. Also, it can be an alternative option for traditional evidence is absent, if it's absent. And also it can support the intelligence of occupancy pattern. For example, EDNA in air can be collected 16 hours after occupancy, and also can be lasted for 3 to 6 months in the indoor area. However, to apply this in forensic field, it requires some significant legal and ethical issues that need to be developed to fully apply this in forensic field. For food industry, EDNA in air can also help to detect microbial contamination, and also it complies with the regulation of Food Safety Modernization Act for environmental monitoring, and also to secure the biosecurity verification of the animal facility. Also, it helped to validate the air filtration system in this facility. Now, if we want to focus on these fields, now the market size of this field has a good opportunity for EDNA to be applied in these fields. For example, the annual growth rate of this market is between 6.5 to 10% annually, with the total amount of billions being spent on these markets. For example, hospitals use around 20 billions, while food and veterinary 26 billions, and forensic field around 2.8 billions, while the EDNA on wildlife markets is around $500 million. So all these can be a good opportunity to be investing in EDNA applications. So now moving to the concept of this technology and how this technology can be achieved. It's like, as you can see, and this is an overview of this is an overview of the technology with the fan that can direct the EDNA air through a filter, specialized filter. This filter can absorb the particles that contain the EDNA, and then after this, we move this filter to the lab to extract the DNA and to do further analysis in the molecular lab. Now, here in King Abdulaziz University, we have succeeded to do this, and we have collected EDNA samples from indoor facility with average equipment number around 45 equipment and optimum operation time between 3 to 4 hours. This has been done in normal temperature, indoor temperature, and relative humidity as well. After this, we collected the DNA in the filter. The filter is specified for capturing small particles, these particles ranging from 0.01 to 10 microns. And because it's a dry system, we can apply this in a very extreme condition, like extreme temperature as well. The diameter of the filter is around 52 millimeters with the correct filters. And we got a good yield of DNA from these experiments, around 0.82 nanograms per microliters, with a total amount of 28.7 nanograms. Now, compared to the actual amount that needed to be done in forensic analysis, the minimum concentration or the minimum amount of DNA that need to be collected is between 0.5 nanograms to 2 nanograms. And we compared this to the literature, we found a better yield as well. We further analyzed the DNA concentration and the quality of the DNA in qPCR lab, molecular lab, and we selected a few genes from the human genes, actually from the human forensic database, and we found that all these genes presented in our samples. Now, moving to this technology, if we want to provide this technology to the field, on these fields, the previous one that I mentioned, so we done a SWOT analysis to check the strength, weakness, opportunity, and threat of this technology. The strength of this technology can be summarized in several points. One of them is that it has non-invasive collection, so it doesn't need direct contact with the sampler. And also, it can be target molecular approach where we can use qPCR for high precision and high specificity. And also, it can be ambient environmental surveillance where we can collect over time while comparing to the other single point collection where for example, the swab surface. And also, we can use the rapid results because it's qPCR, we can get the results in a few hours comparing to the culture. And finally, we can use STR analysis that can unique identification of human presence in the area. However, this technology has some weakness. This weakness can be summarized. First, it has limited validation data because it's a new technology. And also, it has human DNA privacy concerns because it involves DNA. And there's a need for laboratory analysis after collecting the samples. And also, there is a potential for contamination. This contamination can be minimized by strict laboratory practice and controls. Also, there is some challenges for interpretation of the result after collecting the data. However, there is some opportunities to apply this technology, because as I mentioned earlier, that the market size for this field is very high. And also, there is an increased need for infection control after the awareness of post-pandemic awareness. And there is increased demand for regulatory drivers for food and safety, as well as there is the involvement of multiplex PCR, so we can identify multiple targets in a single run or a single analysis. And finally, it has opportunity for strategic partnership with other fields. The threat for this technology can be summarized because it has regulatory barriers due to ethical concerns, and also it could be there's some competitors that can threat this technology, and also the limitation of this technology because it's new and has some, for example, the DNA yield could be low or could be degraded due to the other conditions, but this can be improved by adapting specific lab protocol, and the ethical concern as well because of DNA, and finally the validation requirement for this because it has to be optimized for certain conditions and certain areas. The future direction for this technology can be summarized in 3 points. First, there is a need for developing more robust and efficient sampling and protocols and techniques for collecting EDNA in the air, and also to conduct a long-term study to understand the temporal changes that can direct the biodiversity of the EDNA in the wildlife. And finally, to explore the implication of the airborne EDNA for policymaking and conservation strategies. Finally, I would like to thank the organizing committee at King Fahd Medical Research Center for this opportunity, as well as I would like to thank Dr. Turki Abu Jamal for his input in this project, and my master student, Lujail bin Sadiq Ali Al-Ghamdi. They're a wonderful student, and the center, all the staff at the Center of Innovation in Personal Medicine and the Faculty of Applied Medical Sciences at King Abdulaziz University. Thank you.
Thank you very much, Dr. Majid, for the nice talk and interesting. Now, I would like to introduce the next speaker, Dr. Dua Ismail. She's an assistant professor in pharmaceutics department in Faculty of Pharmacy at King Abdulaziz University. She obtained her PhD from the University of Nottingham in the UK. And part of her PhD, she uniquely combined the qualitative study along with laboratory work. She has research interest in drug delivery and 3D printing. Please join me to welcome Dr. Dua Ismail.
Thank you for attending this presentation. It's just almost the last presentation for today. Today I'll be talking about application of pharmaceuticals and 3D printing. First of all, what is 3D printing? 3D printing is production of an object by addition of layer by layer aided by software. So here we started with creating an STL file through the software. Then we have to slice the object into slices by choosing the pattern and percent and film for printing. There's lots of 3D printing techniques, but those are the mostly common used 3D printing techniques. First of all, material jetting. Material jetting is a very useful technique where the material is deposited through small nozzles, multiple nozzles, and this technique have a very high resolution. Next one is extrusion. Extrusion has 2 types, fused filament fabrication and semi-solid extrusion. The fused filament fabrication is also known as FDM, fused deposition modeling. This technique, we use a pre-made filament that pass through a heated nozzle to form the object. Semi-solid extrusion, powder, and semi-solid gels can be used for this technique. Material jetting and extrusion are the mostly used in 3D printing technique in pharmaceuticals due to availability of materials and ease of use. Third is stereolithography or photopolymerization. This technique is one of the first technique of 3D printing techniques that have been used since the 80s. However, in pharmaceuticals, it's kind of limited due to toxicity of the resin used and difficulty to find the correct materials to use it. However, stereolithography is the build of the object through using a resin, photosensitive resin, that we apply laser to it to solidify the part that we need to print. This type of 3D printing technique used upside down rather than the others, which is building the lower layer then going up. Binder deposition or binder jetting. This technique have been used in the first and only approved 3D printed medicine. In this technique, a layer of powder is applied, then liquid is used to bind the powder particles together. Then the excess powder is removed, another layer is applied and repeated constantly until the object is built. The last here is selective laser sintering, which is similar to binder jetting. However, we use laser to bind the particles rather than liquid. 3D printing have lots of application everywhere in our lives. And here's some examples in pharmaceuticals, medicines, molds, toys, food, furniture, clothes, education, and even 3D printing printers themselves. For today's presentation, we'll talk about the application in pharmaceuticals. Here are a list, and I'll discuss them here. First of all, immediate release tablet. We create an immediate release tablet as a regular tablet, where the dissolution or drug release can be controlled by using variable polymers and ampoule density. So according to the polymer features, we can get what kind of release that we want. Also the ampoule density. So if we create an object with 100% ampoule, the tablet will be compact and the dissolution will be slowly. However, if we decrease the ampoule density, there will be gaps between inside the particles, so the dissolution media will pass through and will release faster. By layer, it can be like immediate and sustained release depending on the ink we are using. So we can use different ink in the same print. Also, we can use a layer without drug and a layer with a drug to control the release as we want. Capsule or caplet, prototyping a body that hold liquid or solid formulation in a controlled release system. As we mentioned before, the release is controlled by using the polymer and the pattern of printing. Polypill, scientists have successfully printed a 5-in-1 tablet, which have 5 separated compartments. 5 separated compartments of to treat hypertension with acceptable size. So we're not using too much materials or excipients, so that we can control, make the tablet smaller. Here they used different release profiles, immediate release and sustained release. For wound dressing, it's been like a personalized wound dressing of biocompatible polymer. It can be synthetic or natural. We can combine it with viable cells to promote healing and proper antimicrobial agent. Also, in this study, they used thermosensitive hydrogel with novel polymer that when it's applied to the body temperature, the bionics will take the place that it put in. We also have implants that have been customized for site-specific physical and mechanical feature. It can be sustained release implant from polymer eluting, so the polymer contains the drug and the implants contains all the drug, or core shell, so the drug is inside the core and the shell without drug to control the release and prevent the burst release at the beginning of the treatment. We also have a microneedle with a biocompatible materials that deliver the dose in a non-invasive manner. Here they used photopolymerization to print cones and pyramid of polymer. Then they injected with insulin, the drug, to promote a fast release of the drug. There are also a floating device designed to float in the stomach with a modifiable floating time, adjustment of the polymer and the internal architecture. So if we have to keep gaps or to make it very compact, so the internal structure plays a role in the water diffusion and floating time. High accuracy needed here because all tablets have to have a unified floating time. Ocular devices also have been printed as a contact lens due to eye limited dose capacity and need for frequent administration. Lastly, here we have a muco-adhesive system. There is a drug delivery system for the buccal and vaginal delivery. It's suitable for local drug delivery and for drugs that have first-pass metabolism. Advantages of 3D printing. The advantages of 3D printing are long to list. However, I have some here. High drug loading, since 3D printing is a single-step development, so there is no need for multiple excipients. So it can go to up to 90% drug loading. Dose flexibility, since we can alter the size and the dimension of the 3D printed tablet very easily through the software, so changing the dose will be very easy. Cost effective, as we mentioned before, that we don't need much excipient, so that will lead to less materials. Also, the material loss during the manufacturing is less. Together with possible use in clinical trials where no need to make very large batches. Better patient compliance, as mentioned before, like in 5-in-1 tablet will be easy for the patients to take them rather than taking 5 tablets to treat a single disease. Also, with the tablets are small and can be printed with a desirable shape that the patient needs, will enhance the patient compliance. On-demand production for the production of drugs with a short shelf life, so we don't have to manufacture lots of medicine and they will expire soon without using, so 3D printing can be an option. Also for delivering medicines for patients in rural areas. Giving an advancement of 3D printing, we can say a 4D printing, which self-change of printed architecture after manufacturing upon exposure to pressure, temperature, water, or enzyme. As mentioned before in the wound dressing, when it's put inside the body, with the body temperature, it changes the printed can swell and take the space that be needed. In conclusion, advancement in 3D printing technology are increasing nowadays. Multiple products are on the way of approval with multiple IND. So multidisciplinary research is encouraged to facilitate inventions for patients' benefits. Thank you.
Thank you very much, Dr. Dua, for this interesting talk. Now, please welcome with me Dr. Abrar Hakami. Dr. Abrar is an assistant professor in the Department of Pharmaceutics at the Faculty of Pharmacy, KAU. She earned her master's degree from the University of Sciences in Philadelphia, USA. Most recently, she obtained her PhD in drug delivery from Cardiff University at UK. Her research focuses on designing new biomaterials using microfluidic devices as an innovative drug delivery system. Please welcome with me Dr. Abrar.
Assalamu alaikum. Thank you, and I'm glad to be here today. Before I start my presentation, let me start with something familiar. I think all of us have used, or most of us have used the rapid COVID antigen test. Did you know that this small chip is a kind of microfluidic system? So microfluidic system has different types. So today I will talk about, I will give an introduction about microfluidic systems and their application in biomedical research. First of all, what are microfluidic systems? As we can see, they are a small device that can manipulate small volume of fluid from microliter to even nanoliters, and then these fluids can go inside the device through micro channels where a chemical reaction or immunological assay occurs. Microfluidics also called lab on a chip. Why? Because we can have all the laboratory equipment in one single device with instant analysis and real-time monitoring. So microfluidic systems started from the 1960 with the idea of inkjet printer and how the ink can flow from small tube, and this was the inspiration to start first lab on chip. Since then, many microfluidic devices have been developed through the years, as we're going to see later on in this presentation. There are several components of microfluidic devices. As we can see in the bottom, this is a schematic structure of a microfluidic device. It has the inlet, outlet points for the entry and exit of the solution, micro channels, also valves, pumps, and detection chamber. Some of these components are optional. It doesn't have to be in all devices like the valves and the detection sensor. I will go quickly for each of them. First, the microfluidic chip itself. It acts as a physical platform to connect all the channels together and other elements. And it's often made from several materials like BDMS polymers, cellulose, glass, plastic material. And according to that, microfluidic device can be classified to either BDMS-based microfluidics that can be used for several applications like in cell or emulsion formation. Then glass microfluidic, they are more chemically stable and can be used for some analytical chemical reaction. Thermoplastic microfluidics, they are cost effective and they are ideal for mass production and for commercial ships. Then the paper-based microfluidic device, as I said, as the COVID rapid test. And finally, the 3D printed microfluidics device. So there are different configurations of the microchannels, as we can see here, either straight line, Y shape, spiral, or even cross junction to form the droplet. And each of these geometry can be used for different or specific applications. And also the inlet and outlet, they are responsible for sample injection and then the collection after at the end. And some samples, as we can see, they can have one inlet and one outlet, and others can have multi inlets and outlet, like the microfluidics at the bottom. It's used for cell separation, and it can have has two inlets, one for mixed cell, the other for buffer, and then different outlets for different cells collection. Then the valve, it's an optional component. It can regulate the flow inside the fluid, the microfluidic device. And it's important for directing or even stopping the fluid if needed. And according to the fluid flow, microfluidic devices can be either continuous flow or droplet-based microfluidics. Moreover, the fluid inside the device can be either passive or active. The passive means the fluid can move through natural forces like gravity or capillary flow, while the active flow, where the fluid needs an external source of energy like pump or electromagnetic field. Then the last component is the detection chamber. The detection unit gives us a real-time monitoring and output measurement of directly on the chip. It can be either optical sensor or electrochemical sensor. The optical sensor detects the changes in light, like in fluorescence, absorbance, or colors. An example of this is in single cell analysis. So the optical sensor can detect the change in fluorescence in each droplet containing cells for cells identification or quantification. And then electrochemical sensor, it measures electrical properties like current, voltage of the fluid due to a specific chemical reaction or immunological assay. An example of this is the glucose monitoring device. It's a type of microfluidics, and the sensor can detect the current change due to glucose oxidation inside the microchannel. So what are the advantages of microfluidics? As we see, they are rapid, give us results within minutes. They have a microchannel, so we only need a small amount of volume, a small amount of samples or reagent. Give a high production rate. They are small devices and portable. The automation that we saw in the valve, so we can control the flow of the fluid inside the device, and they are cost effective. There are a lot of applications of these microfluidics in biomedical research, ranging from disease diagnosis, disease modeling, drug discovery and delivery. I will go through some of them now. First, for point-of-care diagnostic, and the most common example, as I said, in the COVID antigen rapid test. It's a type of lateral flow immunoassay. It contains channels or microchannels made from nitrocellulose materials where immunological assay occur and can be detected through color change. So this is the most common microfluidic use for diagnosis. So they are easy to use, doesn't require any training. They are portable, disposable, And also some of them are compatible with a smartphone for detection and analysis. And here are also some examples other than diagnosis, like biomarker detection, and the most common one, the hormone, also testing, like in pregnancy test device. The second application is in cell culture and tissue engineering. Microfluidic device can provide a highly controlled microenvironment. It acts like a 3D cell culture to mimic the in vivo condition more accurately and precisely than the 2D monolayer, like in petri dish or flask. It contains perfusion chamber, and this chamber allows the continuous delivery of the nutrient and removal of the waste to mimic the blood flow. Moreover, some devices can act as a co-culture system, like the picture here in the bottom. We can use different types of cells, like here different CNS cells in one device to mimic more, or making complex models and mimic the real situation. And here are also some applications in cell culture and tissue engineering, like in stem cell differentiation, or even single cell studies using droplet microfluidics device. So moving from cell culture into organ on chip. So here it's a combination between biology and engineering to mimic the structure and function of living organism. And one example here is lung on chip. We try to mimic the breathing condition to study the disease mechanism, or even some drug toxicity test on it. And if this device can be used as a personalized therapy if the cells were obtained from patient-derived cells. And here are also other organ on chip, like liver on chip, gut on chip. All of these devices, and even the blood-brain barrier on chip, to study some CNS drug delivery and neurodegenerative disease like Parkinson and Alzheimer. And the last application in drug delivery. So microfluidics, either the continuous flow or even the droplet-based microfluidic, can be used for fabrication of drug carrier, like macrospheres, nanoparticles, or hydrogel-based drug carriers. And using microfluidics and by changing some of the parameters of the fluids, like flow rate ratio, the geometry of the microchannels, we can control the size, morphology, and even the drug release of the drug release profile of these carriers. And also, these carriers can act as a sustained and targeted delivery system. And using microfluidic device here or offers a highly reproducible formulation process. In the last 2 slides, I will talk about the work I did during my PhD. I formed the drug-loaded macrocarriers for sustained local delivery to the brain using a droplet-based microfluidic device. So first, for formation of an emulsion, then using a specific technique to convert this emulsion into a new type of biomaterials called cryogels, and then use these cryogels as a sustained local drug delivery system. Here I used a commercial microfluidic chip, as we can see, and we have 2 inlets for oil phase and water phase connected to the syringe pump, and on the right side the outlet for the emulsion collection. As we can see, we got monodisperse uniform particles of an emulsion with high production rate, and it was very highly reproducible. Then I convert this emulsion with specific technique into these macrocarriers. It's like a spongy-like material, and it showed high drug loading efficiency. And then we used this material as implanted, as injectable implant to the brain. And as we can see, they were able to slowly release the drug at the injected site. So finally, the challenges of microfluidics now, they're trying to first make the human on chip that use multi-organ in one chip to mimic the human body. Then scale-up production is still challenging as well, as it needs a lot of standardization. Also, we can use the 3D technology to make more customized microfluidic device for a specific application. And finally, AI integrated microfluidics. So we can, the AI algorithm can help us to optimize or even feedback of the data for more complex microfluidic devices. So to conclude, so microfluidic devices are fast, precise, and cost-effective. They are more than a tool. They can be used in several biomedical applications, from diagnosis to more regenerative medicine. And they will continue to bridge the gap between lab research and clinical translation. And thank you for it.
Thank you, Dr. Abraham, for this amazing talk. Now we will start the Q&A session. For all the speakers, please join us in the Q&A session. Thank you for the amazing talks, doctors. And I think I'll start with Professor Ahmad. Regarding the smart health applications, so what is the level of impact of these applications on the smart health applications in our community?
So, thank you for the question. At Counts, the example I gave are the results of many years of work and support from funding, mainly internal fund, both the research, the support for the research from the university, but also from programs like from Smart Health. Now, we are thriving of all of them to move at the commercial level. Not all of them are at the same stage. And the difficulty we are facing, in spite, COWS, of course, have the means, have the support, the logistic, we are still struggling, finding ways to move our finding, the ones I've introduced, I presented, but also others, to the market. For example, one of the challenge is access to patients. That cost has been struggling with it. We have partners, we talk, we have some of them being tested. For example, during COVID in many ways, it's weird to say it that way, was a blessing because doors were open fast. For example, for Samir Hamdan, he tested very quickly his kit and validated and moved to SFDA. We have a lot of discussion, and this is where now the center is making a lot of effort to build partnership with hospitals. Faisal, of course, is one of our partners, but also there are others. Humanitarian city in Riyadh, Keyma, National Guards, and we are hoping closer to home, Jeddah. I spoke last week to IMC, now with Dr. Nazim, with Faqih, etc. We are willing to work with hospitals, with healthcare institutions to test our devices. Of course, we will work with SFDA and policymaker for the regulation, but also that's our objective. So many of them are ready to go, and many of them are ready to be tested in patients and move to the next level. So that's where we are, and we are hoping next time, if I come, you invite me back, I will give you better news. Yeah, absolutely.
For Dr. Hamza, regarding the DNA-based diets, how DNA-based diets works, and how do you think they are applicable for the Saudi community?
I believe that this platform, when they first analyze or sequence our gene, the gene of the client, they search for polymorphism or for polymorphism for digesting carbs or digesting, or how the people are sensitive for caffeine and different or lactose. And then they will design or tailor diets depending on their specific condition. However, later on, especially in here in Saudi Arabia, this aligned to actually to be applicable in Saudi Arabia. Actually, these diet plans can be designed and tailored to our local food, also our local way of living, like we fast in Ramadan. And as well, actually, this aligns with the vision 30, where there is tailored, personalized prevention are coming with this diet plans.
For Dr. Da, regarding 3D printing technology, how do you think, or when do you think we can come up with personalized therapeutics using 3D printing technology?
The main problem here is the regulation. So there is no guided regulation directly for medicines. There are lots of regulations and medicines. So like devices and something like that. But for medicine, it's strictly guided. So that's why even the only approved drug is manufactured in a big manufacturer. So not a 3D printing. Maybe the personalization would be like in a small, like in a hospital or a pharmacy, that would give the personalization. However, manufacturing in big manufacturers with large bunches, it's not that what we want from 3D printing.
Like we for example, with Dr. Salimban and Dr. Walid, we came up with a 3D printed tablets that involve 3 multiple medications. So all of these medications are really approved for clinical use. But what do you think about the challenges or the problems that might come up with combining these drugs in one tablet? So
I think since researchers did the 3-in-1, they know that it's compatible, that they are compatible with each other. The main problem is that we can ensure that all tablets have the same quantity of the drug and gives the same effect on all patients. So if we have much research on 3D printing that can get the regulatories and even pharmaceutical companies to trust 3D printing more, and that will give regulations that will help to have them in the market.
Thank you, Dr. Dr. Abrar, just regarding microfluidics, I worked with the microfluidics for generating emulsions or nanoemulsions, and the beauty of this technology is to give more dispersed products or formulations. So, what do you think is the most applicable or the most potential application of microfluidics in the medical field?
So yeah, I use microfluidics for an emulsion formation. So it's mainly to make a monodispersed emulsion. Microfluidics itself can also be used to make nanoparticles and microspheres and all this formulation. The idea of this formulation to obtain the monodispersed particle size, and this is very important for this is important for drug release profile of these macrocarriers and how we're gonna test them later on.
So, just the question is that, like, I know the other technologies like high energy methods, they produce also drug delivery systems and nanoparticles, and I know that, like, in the pharmaceutical field, they always worry about the variabilities in size and physical parameters in general. Do you think microfluidics might overcome these problems in future, like in generating nanomedicines, for example, or lipid-based formulations? I think Pfizer and Moderna were produced using microfluidics or other techniques. What do you think?
The Pfizer or
Yeah, they are also lipid nanoparticles.
yeah, I think so. Yeah, so the microfluidic is the potential now application to make different macrocarriers for their monodispersity. So yeah, using other technique to formulating these macrocarriers, as you said, or spray drying or different kind of these techniques can still have some variation of the characterization, while microfluidics like give us a consistent particle size and highly reproducible. Some of these technique doesn't have the reproducibility.
Thank you. Dr. Majid, just regarding the DNA capturing technology of viruses, how do you think this technology is compared to other surveillance technologies in controlling infections in Saudi Arabia?
It's a promising technology actually, because it's a rapid technology compared to the traditional way. There is other technology that can be also used to collect DNA from the air. However, these technologies depend on other factors. For example, propagations that need filtrations to be installed in the machine for collecting bacteria, for example. So this is the dry system. It has no honey liquidness, can be optimized for extreme conditions, but temperature. Also, it can be collected for both. Actually, this is the first step that we collected DNA. We are aiming to collect RNA for viruses later on, and hopefully we can get, it's very difficult to collect RNA from the air, so hopefully we can get it.
Thank you, Dr. Majid. I think now we'll give a chance to the audience if they have any questions.
Yes, I have a question for Dr. Abrar. So you mentioned that microfluid used to drug delivery. Can the nano carrier be designed to deliver more than one component, each targeting different biological pathways?
Sorry, I didn't hear you. Can you?
Yeah, sure. Yeah, I just asked if can the nano carrier be designed to deliver more than one component, each targeting different biological pathways?
Oh, I think I haven't seen this thing, because if these particles can use as implant, as injected or as local delivery system. So So if you mean by your question to use different drug or components inside of these to act on different sites, I think it's localized therapy, so we can use one type, but these two therapy can be act on the same site of injection. I think that's okay, but if it's different location, I don't think so.
Any other questions, Dr. Khalid?
Thank you for all speakers for these interesting topics. I have two questions. For the first one for Dr. Majid. Are there special consideration for extracting DNA from the filter? Maybe highly contaminated, for example, special kit. And for Dr. Abrar, what are the specialists that should work together to make a success microfluidics project, in addition of medical field? Thank you.
I'll start for a second. Okay, and that's actually a good question. For contaminated samples, there is a very specific protocol. For example, if you want looking for human DNA samples, then you need to extract human DNA. This depends with the application later on. For example, we can do sequencing if you want to analyze the data. And then you can check, for example, let's say for forensic field, for example. If you're looking for someone who's been present in a small room, for example, then you need to detect the stem regions for specific genes and then identify these genes around. Actually, in UK system, I think they use 12 genes. Then after this, you can obtain the same samples from the suspect. Then you can compare these samples together. If there is a match, it should be there is a match. Like I mean, STR region usually they don't match more than 8. So it's a 12 genes should be fine. Yes. So this is one of the solving these problems. Also, you can optimize the protocol to get a better yield without degrading the DNA. The technology that we use, it's a foam technology that can extract the DNA from the filter without damaging the DNA. Hopefully I answered your question.
Sorry, so your question was what are the standardization to make the perfect microfluidic device? Am I right?
أقصد التخصصات اللي ممكن تجتمع بالإضافة للتخصص الطبي لنجاح مشروع.
Medical or engineering, because it needs how to fabricate the microfluidic. I think it needs a lot of engineering with medical field as well.
Any more questions from the audience? One here last. Please.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you so much for the great panelists today. We have enjoyed your talk, all of you. I have a question for Dr. Abrar. Regarding your insightful talk about microfluidics, I would like to ask about the real-world practice in our country and in our institute here at the university. If we have any advancement or do we have any availability of organ-on-a-chip technology? For example, for any potential researchers for background of cardiovascular medicine, heart on chip, or like blood vessel on chip. Thank you so much.
Thank you for your interest. Actually, to be honest, I just came recently from my PhD, and I'm more than happy to start microfluidic here, research in the university. So I didn't start it yet. So if I do, I will be more than happy.
Okay, thank you very much. Just a question for Professor Ahmed.
From strategic point of view, what is your strategy and the position of the gut microbiome in frame of the human smart health?
So, studying microbiome is really at the heart of the One Health theme. We have two colleagues. I think one of them who presented here, Dr. Arnab Payne, he's working on microbiome, but also Dr. Danish. He was recruited 2 years ago to develop the arm and axe of microbiome in human. Our strategy is to study the microbiome in the linked to diseases. So, and also, ideally, is to study the adaptation of microbiome in response to environment challenges and environment changes. And to me, I mean, where we are heading with AI integration and AI data mining, if we can integrate the microbiome from what is inside the gut and also the changes that are influenced from outside as in response to environmental challenge, in addition, of course, of the genetic background, etc., etc. So all of that, that's where we are heading, and we are hoping to bring to Saudi Arabia through this research that microbiome become standard of care for diagnosis to follow up development of disease, like diabetes, for example. We know that there is a direct connection, etc. That's the thinking and the strategy.
Okay, thank you for all the speakers, and thank you for the audience for listening to these interesting talks. Now, very much.
خذوا جروب فوتو دكتور او نفي دكتور دكتور حسام
Let's go to of the week. That's good.
والله دكتور
Now we will announce the poster prizes. We will announce the top 3. So rank number 3 is Tamar Cindy. The second place is Hadi Al-Mahdi. And the first winner is Reham Al-Mukhtar. Congratulations for all the poster winners. And we will conclude with Prof. Anwar, the conference.
السلام عليكم. يعني كنت كاتب كلمة صراحة و I don't think I'll read it all, بس يعني I'll try. It will be in Arabic, so forgive me if you don't. I can't translate it. طيب بسم الله الرحمن الرحيم والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد عليه أفضل الصلاة وأتم التسليم. الزملاء والزميلات الأعزاء، الحضور الكرام، في ختام مؤتمر مركز الملك فهد للبحوث الطبية الأول 2025 لا يسعنا إلا أن نعبر عن بالغ فخرنا واعتزازنا بما تحقق خلال هذه الأيام الملهمة، والتي شكلت منصة علمية استثنائية لتبادل المعرفة وتعزيز التعاون واستشراف مستقبل البحث والابتكار في مملكتنا الغالية. على مدى 5 أيام وتحت شعار تحسين جودة الحياة من خلال البحث العلمي والابتكار، استعرض المؤتمر أبرز المستجدات البحثية والعلمية في محاور متعددة شملت الأمراض المعدية، التصوير الطبي، علم السموم والعلوم الجنائية، علم الأورام وأمراض الدم، الأمراض العصبية والشيخوخة، التقنية الحيوية والابتكار. لقد فاق الحضور جميع التوقعات، يتجاوز عدد المشاركين في المؤتمر ما بين سواء بالحضور الشخصي أو عبر الأثير أو عبر الإنترنت البث المباشر 5000 شخص، مما يعكس حجم الاهتمام الكبير والتفاعل الواسع مع محاور المؤتمر. شهد المؤتمر مشاركة أكثر من 100 متحدث محلي ودولي فيما يقارب 19 جلسة علمية متخصصة، إلى جانب نحو 300 ملصق علمي من قبل طلاب وباحثين من مختلف التخصصات ينتمون إلى أكثر من 60 جهة محلية ودولية، مما يعكس أهمية الشراكة والتعامل في التكامل في دفع مسيرة البحث العلمي نحو آفاق جديدة. وفي ضوء ما طرح من جلسات ونقاشات علمية، هناك بعض التوصيات التي خلص إليها المؤتمر، ومن أهمها أهمية بناء شبكات وطنية متكاملة للبحث العلمي، تعزيز التنسيق والتكامل بين المراكز البحثية والجامعات والمستشفيات، تعزيز التعاون بين الأطباء والباحثين لتحسين الرعاية الصحية، تسريع تبني التقنيات الحديثة مثل الذكاء الاصطناعي والطب الدقيق في التطبيقات السريرية، تمكين الباحثين الشباب والطلبة عبر إشراكهم في المشاريع البحثية وتوفير برامج تدريبية نوعية، ترسيخ ثقافة ترجمة مخرجات الأبحاث إلى تطبيقات عملية وربطها بالسياسات والاستراتيجيات الصحية والوطنية. وبهذه المناسبة نرفع أسمى آيات الشكر والعرفان إلى مقام خادم الحرمين الشريفين الملك سلمان بن عبد العزيز آل سعود، وسمو سيدي صاحب السمو الملكي الأمير محمد بن سلمان بن عبد العزيز ولي العهد رئيس مجلس الوزراء حفظهم الله، على دعمهم المستمر للبحث العلمي والابتكار وتعزيز مكانة المملكة في هذه المجالات الحساسة. كما نتوجه بجزيل الشكر والتقدير إلى سعادة رئيس الجامعة على رعايته الكريمة لهذا المؤتمر، وإلى سعادة نائب رئيس الجامعة للدراسات العليا والبحث العلمي على دعمه ومتابعته المستمرة لإنجاح هذا الحدث في رحاب جامعتنا الحبيبة. ولا يفوتنا أن نعبر عن امتناننا العميق للرعاة والشركاء الذين كان لدعمهم الأثر البالغ في نجاح هذا المؤتمر. وهنا لا يسعني إلا أن أوجه أصدق مشاعر الشكر والامتنان لأبطال مركز الملك فهد للبحوث الطبية من موظفين ومتدربين وباحثين وطلبة ومتطوعين وأعضاء لجان علمية وإدارية وتنظيمية الذين عملوا بتفاني وإخلاص خلف الكواليس لتحقيق هذا النجاح المشرف. أخيراً نجدد شكرنا وتقديرنا لكم أنتم ضيوفنا الأعزاء ولكل من شارك وساهم في هذا الحدث. نأمل أن تكون هذه الأيام قد أثرت معارفكم وفتحت آفاقاً جديدة للتعاون والإبداع والتميز. نلقاكم بإذن الله في النسخة القادمة من المؤتمر، والسلام عليكم ورحمة الله وبركاته.